Good morning, everyone. I'm Ananta Shaker. I'm the Senior Vice Chancellor and the Dean of the School of Medicine here at University of Pittsburgh. And it's really a great pleasure for me to welcome all of you to this symposium, From Space to Earth, Biology as a Bridge. We are trying to put together here a concept of what we know today of medicine and biology on Earth, and what we have learned so far about biology and medicine in space. And with that initial knowledge, we see that there is tremendous opportunity to bridge the two. We can understand some of the foundations of our biology, our creation, if you will, by actually understanding what biology is like in space, in, I would say, extraterrestrial worlds, and presumably in conditions that are not normal for most Earth atmosphere living creatures. So, and why is that exciting? Well, it's exciting for many reasons. The first, I think the most important reason is that we as humans have always wanted to be in space, in the understanding the bigger picture of universe, origins of life, and how our foundations of our carbon-based biology has been created. And we can't do that without understanding other types of possibilities or even stressing our existing biology to non-terrestrial environments. So I think it's a fundamental question of biological research, is that what would biology be like if you're not in this environment? And how is that affected in terms of cellular and subcellular functions? Because I think that would give us better insights into how to improve or how to understand our current unknowns in the biology. The second thing that we really want to think about is, as a leading academic institution and as a leading biomedical research institution, we also want to be prepared for the 21st and 22nd century humanities needs. Let's see, I'm already being an optimist. We're going to be in the 22nd, we are going to make it to the 22nd century. And when we do make it to the 22nd century, we will probably be not just limited to terrestrial lifestyle. And with that, we can't be actually, I think, physically, we'll have to start thinking about extraterrestrial environments for us to successfully make it to the 22nd century. But what that means is that we have to be prepared to think of medical care, to think of biology, to think of engineering solutions in very different environment. And that's what you will hear today. We have an amazing lineup of speakers, starting with our headliner. Kate, oh, wow, you've got your NASA uniform and everything. So, Kate's almost all of you would have heard of her. She was not only an astronaut who spent multiple months in space, but for the first scientist to sequence a genome in space, in zero gravity, which means that she had to create an amazing amount of technology to be able to do that in zero gravity, in limited space, with limited power, and I'm sure you'll hear more. But that's just the beginning. Then we have variety of extraordinary national and international leaders who have done space biology, space research, and will hopefully be involved in many of our future activities. But all of this wouldn't have happened if I hadn't actually recruited this super energetic, super positive, hyper space biologist called Afshan Bhatti. Afshan has been the catalyst for a lot of this activity. He is a director of our Space Medicine Center and the deputy director of the McGowan Regenerative Medicine Center. And he gets full credit for opening my eyes to possibilities of space and future and where Pitt Health Sciences and Pitt as a university can claim a unique presence for the next century. So with that, I want to call up Afshan, our sort of conspirator for all of this, and thank him again for his great work and bringing together this amazing star of space research. Thank you.
Well, thanks. Anantha's too kind. So I'm going to quickly, so you don't want to hear me talk, you want to hear everyone else talk, Chris, Sylvan, John, and Sarah, and Danica. But I'm going to give you a quick recap and overview of what we're doing for space. We've been in the news for space biomedicine. Chris and I were actually in the New Yorker article, which I'm sure Chris is going to talk a little bit about. And this is a good article, actually get your bearings in how space research is done, from all the neat things that Chris is going to talk about to what we do here to all the travel happens after that. There's also a caucus in Pennsylvania. So Pennsylvania's Lieutenant Governor has launched a new aerospace caucus, which I was part of a panel session, which more to come there. So we do have the backing of, not the backing, but the governor's office is very much in Pennsylvania into space research. So that's a key program, the key thing that I think is really beneficial for people in Pennsylvania and Pitt. And of course, there's a magazine out there, our Pitt Med magazine, we talk all about our program here, so pick that up on the way out. So yeah, Pitt Space was started, originally it was Alan George with space engineering, and then Michael Ramsey, and then I came to do space biomedicine, as Nantha said. And we covered a lot of different topics and more, basically everything under the sun related to space biomedicine. So it's going to be fun to do a lot of different things. So what is the future initiatives in biomedicine? One thing we're trying to do is starting a whole global education outreach kind of program, certificate program, where quickly a bunch of universities and agencies, and working with Chris and other folks to try to set this up, where we're going to harmonize the whole education of space biomedicine around the world. And Pitt will be the hub of that, of course. So this will be a whole online program. Another thing that Chris and I are trying to do is do a whole joint PhD program, or hopefully other universities to do that. So maybe stay tuned in the next year or two to hopefully do a whole joint space biomedicine PhD program, kind of first of its kind. But we'll see how long that'll take. Hopefully we'll get that going up. But you're all here to see what's, I'm not going to talk too much about research because we got all the great people like Kate, Chris, Sarah, Sylvan, and Danica to talk about space research. But one thing, why care about space? The main thing that people care about space is that space is an accelerated model for a lot of diseases and aging. Other than the fact that it's fun to go to space, well, for some people, not everyone. You're going to hear Kate and John talk about why it's fun. So, but the other part is why it relates back to Earth and the clinic, because it accelerates a lot of models of aging, diseases. Mitochondrial disease was one of my passions. So that's when you hear the talks from everyone, that's the theme that you want to make sure to keep in mind, that everything we're doing in space really impacts what happens on Earth here. And the other part is basically it's a booming industry. It's estimated to be about $2 trillion, and then it's already happening in the next 5-10 years. Space research is going to be that. And then there's also the whole idea of space tourism. Can you go, instead of going to the Hawaii for vacation, maybe you want to go to a hotel around the Earth. But we have to make that safe for people to travel first. So, this is the goal. We want to make people like Ardeen hopefully be able to go to space. And that's the key, safely. That's what we want. So hopefully not only Kate or John gets to go to space, but hopefully Anantha could join us there. But that's the whole goal. Maybe everyone here can be safely traveling space, and Chris and everyone else here will be talking about ways to create the magic pill and make it safe for everyone to travel to space. So without any further ado, because you're not here to hear me talk, you're about all the great people talk. And there's a whole new initiatives that are happening with Vishnu, Dr. Vishnu Vardhan, who's going to talk about all the great PIT, VISI, Longevity Lab, and how that's going to be involved with the whole space research. So I'm sure many of you know Vishnu. You have his talk. Okay, good. So without further ado, I think Vishnu, you want to talk about how VISI is part of this and how everything you're doing, great things you're doing here. So thanks everyone, and it's going to be a fun day. You see me come up and down, up, back and down to introduce people.
That was for you, not for me.
No, all for you.
Yeah. Good morning. It's great to be here. What has VISI and Multi-omics and our initiative got to do with space? Before that, it started like 2 years back when Dr. Shekhar came to Bangalore to inaugurate our Bangalore office. And at that time, we talked about how we can use multi-omics to understand biology, and then can we translate it in real world? Is even multi-omics useful in real world, or it is only in the research? So in 2 years, what has happened? What is it? Yeah. Sorry. Okay. Okay. It's fine. So today, I want to talk about two things which we are initiating here. The first thing is the multi-omics lab, which we are setting up. It will probably be by the end of August. We are expecting to start. It will be world's first multi-omics lab, the largest one. Currently, we have a lab which we run in Turkey. This will be like 5 times bigger than that. And so what this lab will be doing is anything from whole genome sequencing to 11,000 somologic proteomes. So we are planning to do all 9 omics, whole genomes, metagenomes, transcriptomes, then epigenetics, lipidomics, microbiome, and proteomics, the same place, parallelly, where a single sample, you take blood, stool, urine, saliva, you can run through this and then virtually get all this. So this is the wet lab part of it. The amazing thing which we are also doing is we have built a bioinformatics panel which can take the raw data from all this and then create panels, which I'm showing today. What I'm showing is currently what we are going to launch in Austin. It's world's first CSC reversal program. So this is how the multi-omics architecture we have designed. You can take anything from genomes, epigenomes, transcriptomes, proteomes, even real-time sensors, from anything. We have integrated 150 sensors, any sensor in the world, CGM, smart ring, watch, HRV, any sensor. And then we create this multi-omics architecture. And this is our AI. Today we call it as Codex AI. So what it does is, it creates biotypes. So many of the words which I am talking today are totally the first time we are using. These are all, which we have started using. So the biotypes, I will explain like what we are going to do, and HPRX. So this is the multi-omics lab services. So you take blood, stool, anything, and then we can create this panels. It's a CLIA certified, CAP certified lab. Coming back to the CAC, which we are launching, it's going to be world's first CAC reversal program, because currently, what happens if a person is having a CAC positive score? So we get treatment mostly, okay, you get statins, you get aspirin, and then we wait for them to get a block and then stent it. So we are doing something totally different. So we started with diabetes. In fact, Vizi started as a diabetes reversal company in India. So we started working on diabetes reversal. And then now, this is how it happens with CAC. So we create biotypes based on the multi-omics panel. And then the intervention starts based on the biotype, which is first understanding the biology and then precisely managing the care rather than. So this is how, this is not, we use a lot of AI here, and this is not generative AI. It's totally, we call it as biological intelligence, because the AI here does three things which current AI doesn't do. So number one thing it does is AI has memory. So if you look at all the LLMs and chatbots which we are using today, they don't have memory. You can start talking to them, and tomorrow you come back and start, so they will again start from the beginning. They will not remember anything. So this is one of the major factors which will affect AI being used in clinical care, because if there is no memory, it's going to be disastrous. That's what they call it as hallucination. It's not hallucination. It's the sheer fact that these LLMs have advanced artificial amnesia. They can't remember. They're just pattern seekers. So you give a prompt, and tomorrow again they will see the same prompt, and then they will seek a pattern. They will not remember what they spoke to you yesterday. So this is the biggest challenge, to use generative AI and LLMs in clinical care. So it needs memory. So like tomorrow we come back, the LLM or the AI should remember what has happened with us today. So that's how the clinical care works. So this is one problem which we have really sorted out here. The other important factor is, in healthcare, is something which is very, I can call it as the greatest innovation which we have done on AI. That's the SCAR. SCAR is something, we go to a doctor or else a patient, so they have a lot of past history. We just call it as past history in our clinical care. But past history is not something which we just note it on the notes, but it is in the brain and it's in the memory of the person. Previous interventions which have failed, what has happened to their father. If you take my own history, so I have 4 generations of very bad diabetes, which is always there in the back of my brain, that it can affect me also. So this is a scar. Currently, no healthcare system takes this into the consideration. Like how this is affected, like the previous medicine, previous failed treatments, what has happened to this person, that is not remembered. So that we are taking into consideration. And most importantly, what is happening is, when we advise precision care, so people usually take, I advised, but they didn't listen, like it's very difficult to change behavior. That's the biggest challenge. I meet so many insurances, doctors, payers, everyone says, changing behavior is the impossible thing, because nobody wants to change. Nobody wants to quit their sodas and, Pepsis and their thing. So you can't change the people. The best thing is to just, medicate them, give them GLP-1, and so it will reduce their weight. So that's the easiest thing we can do, because people are only taking medicine. They don't want to change. But the fact of the life is, why people don't change, why behavioral change is impossible, is because we have divided behavior into mind and then body. What we are combining here is, it's not like that, you know. Now, why people don't change is because of the fact they are not ready to change. It's in their metabolism, it's in their mitochondria. That's why when Afseen spoke about a mitochondria, I said, this is the team I want to work with, because I will tell you why. Many of the behavioral changes that I see in my cases, where I am saying that we reversed so many things, that happens because of the fact that their mitochondria mostly is not ready. Most of the mitochondria is being pushed as mental health and all the other problems, but it's really the mitochondria which doesn't affect, which doesn't let people change. And that's what we are really understanding with the epigenetics and then trying to change it. So currently we are launching this, and there are a lot of other programs which we are planning to launch. One of the most interesting thing which we want to do is space twin simulations. I have built a simulation engine of like nearly 1 million biotwins, and we want to do that for organs and for space twins. So that's the program which we are very interested in. Can we build simulations of different organs, send it to the space, understand what happens? How does the liver behave there? How does the heart behave there? It's going to be amazing. So this is one of the projects which we'll be undertaking. Sylvian has proposed that, and I'm sure he's going to talk about it. And another important thing which we are going to work is the mitochondrial pacing for fatty liver. I call it as biotype 10, where we are going to create this biotype 10 based entirely on the mitochondria. And another thing which we are planning to launch in India is, India is seeing a lot of sudden cardiac deaths. People are blaming it on COVID vaccine. Like yesterday, there was, you'll not believe, a fight between 3 of state chief ministers and central health minister blaming COVID, where virtually the whole biotech industry got involved. Suddenly, a lot of very, celebrities have died because of the cardiac disease, and everyone is saying they all took COVID vaccine. So it's just like, it's just taking medicine to entirely different place, like where people are fighting for something which has saved millions and millions of lives. Today, if I'm standing here without having any limp, which was very frequent in 2 generations back in India because of polio, you don't want to kill that, right? So it's so scary to see that people are pushing because they don't know why this happens. So it's easy to blame something which is already there. So COVID vaccine is the only thing which has changed in the last 4 years, and these people are dying because of that. So it's so easy to just blame that. But India has huge problem of sudden cardiac deaths because of 2 reasons, which we found out in our cohort. 1% of Indians have channelopathies, and nobody knows it. We don't look for it in our genes. So channelopathies can cause sudden cardiac death. 1% Indians is nearly 14 million people. It's not a small number. So these people, this is one of the reasons. The other reason is, if you look at the pollution of India, it's always super polluted. Delhi, Bangalore, wherever we see this are also polluted. So how is pollution even affecting our cardiac? We don't know that. So instead of looking at the real reason, we are just saying, okay, COVID vaccine people are taking, and then the protein spike is killing people. So, which is totally, nobody has proven that. It's just people are talking about it. So that's the program which we want to launch in India. It's going to be as PS protocol, where we'll be looking at heart organ, how pollution is affecting it, what is happening with sodium channelopathies, is it causing sudden cardiac deaths, make a different biotype for Indian population, because our India is 1/6 of world population, and a lot of Indians have not been studied. All the SNBs and PRS of India's are mostly taken from UK Genomics report. So we are going to launch next lab in India, which is will be even perhaps, I think my brother is working on it, bigger than what we are setting up here, because India is 1.4 billion people and we have only a 10,000 gene panel to understand Indians. It's so small, it's 0.0001%. I don't know, we have not studied Indians at all. And this is something which I'm very keen to take our whole space research there to India, to ISRO, which is India's. ISRO has done amazing work. If you know space, like we have sent moon mission for like less than gravity movies budget, which was a movie, and we sent it for the lesser money. So Indians have this knack of doing things for a lot less money than what it costs. This is another example how we have done it without raising. If you look at the whole AI, Grok is spending $1 billion a month, and we have done it in much, much less. So that's the knack of what we are doing. So these are four initiatives I just wanted to talk. The important thing is this lab which we are opening, this is something which gives huge advantage in understanding the multi-omics, because this kind of scale is a very unprecedented. No lab in the world has this kind of scale, and most importantly, the bioinformatics panel, which can really, I have totally worked with NVIDIA on their chips and directly build this, which makes it very easy. Like you can get reports in, like we are looking at hours, not even like weeks or days. In fact, we wanted to try it like real time, but my Adil was supposed to come today. He missed his flight. His flight got cancelled. Otherwise, he's my partner who will run this lab, but he's not going to come. I think he'll talk online. So we wanted to do it in 5 minutes, but Adil was like, why to spend 10x GPU cost on that? Can we do it in 2 hours? Whole genome sequencing. So that's where it's really come. The whole infrastructure to do this will be set up in Pittsburgh. So Pittsburgh will have world's first multi-omics lab. Pittsburgh will have the entire AI GPU infrastructure, the whole AI. So we are thinking Pittsburgh should become Framingham 2.0. That was for the heart, but this should be for the biology. That's the vision of GainMed, which Dr. Shekhar had, and I'm trying to, put some effort to enable that. When he told about the vision, I was like, wow, this is, how do we do it? But now I see that it's happening, and it's happening at a very fast pace. And this whole space research initiative, which happened in this last 2 months, is an example of how things can go really fast here in Pittsburgh. And I'm really looking forward to do many more initiatives which we are planning in trauma, in other cases. We'll really be launching a lot of projects here. Thank you very much. It was amazing talking to you all in the morning, and then give some heads up on what is happening in Pittsburgh. The AI, the multi-omics, and the whole infrastructure we are trying to bring in here. Thank you very much.
Thanks, Vishnu. I guess we'll have questions for him at the session for the break, right? So I want to introduce Kate Rubens next, Dr. Kate Rubens, who is, I don't think any introduction is needed, but she's a NASA astronaut, but she's also done many scientific research and many different important work throughout the years. And without any further ado, I'm not going to talk. And so for the speakers, I'll stand up when it's like 5-minute mark, so then you'll see me. But yeah, come on up. And then we'll have questions at the end too. So there's 2 mics. There's 1 mic here if you come up to the questions. So let's welcome Kate.
Thank you so much. So I'm super excited to be here today. We've been planning this for a little while, and I think the collection of speakers that Afshin and Arantha have assembled here is really incredible. It's going to give you a sense of the depth and the breadth of space biomedicine and space research. And so I wanted to focus on some of the work that we've done over the last 5 years on the International Space Station and give you a little bit of a flavor about where we're thinking, where we're going to go next. And so my talk's centered around advancing biomedical innovation and how we're actually using space to push us forward in some really interesting ways. And is this, all right. So just take a deep breath and picture yourself here. This is looking out the window of the cupola, looking at the space station, and you can see the Earth. You would think the Earth would be below you, but because your head's down in the cupola, it looks like the Earth is above you. And you can see this is winter on the planet, right? You can see the landlocked like ice flows. You can definitely tell that this is a season where there's a lot of ice on the planet. And just picture yourself going along at 17,500 miles an hour. And you're looking at this beautiful, incredible blue marble behind you. Up here, gravity's just a whisper. We call it microgravity because it's almost undetectable unless you're doing a very sensitive physics or material science experiment. For biology, it's essentially completely undetectable. So cells float. They don't settle to the bottom of a petri dish when you put them in a petri dish. Biology behaves in ways that no earthbound lab can replicate. And so this is what I want to talk to you about today, because when we lift that weight of gravity off of these living systems, we can find entirely new pathways for discovery. We can improve our understanding of life back on Earth because we can find insights into things like cancer cell growth, regenerative medicine, mitochondrial behavior, virus reactivation. These are just like a handful of things that we see changing when we no longer have the pervasive influence of gravity. But first off, how did I get there? So this is me, and stuffed, and I mean stuffed. Like the technicians that load us into the capsule, I feel like they're gonna get their foot out. Like when you're packing a suitcase and you really can't get it closed. That's what it feels like sitting in a Soyuz capsule. So they packed me in first. That empty seat next to me is my commander who's taking this picture before he gets in. And then after the three of us get in this really roomy capsule, they pack more stuff on top of us. But this, I kind of felt like this is one of those things, if your kid's ever done that experiment where they build something around an egg and they drop it from a building and they see like, well, you know, what's good cushioning? I'm like, I feel like we're pretty cushioned in this capsule. So you get in this capsule and then you've got about 4 to 6 hours before launch. And you sit there and wait and think about all the bad decisions in your life that have led you to the point of sitting on top of what's essentially an intercontinental ballistic missile that's ready to leave Earth in a controlled explosion. There's zero chance of backing out at this point. It only takes 9 minutes to get to space. So you accelerate from 0 miles an hour to 17,500 miles an hour in 9 minutes. And this is our destination. So this is the International Space Station. This is humanity's most incredible engineering feat, I think. This is the size of a 747. With the solar rays, it's as big as a football field. And humans have built and assembled this engineering marvel in space, piece by piece, over almost 25 years. So when we're up in this space station, the reason that we're there is for science. We are there to look at everything from biology to fluid mechanics to physics to Earth observation. That's what we're doing. We spend some portion of our time doing maintenance on the space station, but almost all of the astronauts' time is devoted to science. So I did over 200 different scientific experiments while I was up there. It's incredible for somebody with like a little bit of ADHD, the fact that you can go do 200 different scientific experiments in fields all over, the things that I never would have learned before, and work with PIs, internationally recognized scientists from all over the world. This just makes the space station an absolute amazing marvel for scientific inquiry. And you have this ability to essentially get rid of gravity. So all these experiments that we're designing for the space station, as you can see, it's a little bit different, the materials that we have on the space station, right? This does not look like your normal lab. So everything has to be designed to fit in the size of a shoebox. We have to think very carefully about how much power every experiment draws. We have to ensure that it's robust, that it can survive launch loads. We have to carefully think about how it integrates on a systems level and a software level with every other piece of equipment. It's very different than designing equipment for research on the ground. But when you design hardware and software for space, by the time you finish with that design process, you've designed something that's modular, remote, robust, rugged, interoperable, and easy enough for an astronaut to operate. So think about where you could use that kind of hardware on Earth. I'll talk about this a little bit more later, but things like rural health clinics, going out and looking at global health in other countries. We've developed this system where we build a giant hospital and we ask the patients to come into the hospital. And that's where they get their samples done and their blood taken and their medical analysis. But think about how many more people we could serve with some of the incredible technologies that Vishnu was talking about, the genomics, the epigenetics, if we had methods to collect samples and analyze them. And I think this is one of the things that's maybe not recognized about space, but it forces your design thinking to make small, portable, rugged hardware. And so I think that the hardware that we're going to develop for the space station is going to affect every place on Earth. So why microgravity? We talked a little bit about this, right? This is this unique laboratory for scientific discovery. And it reveals these hidden processes that are masked by gravity on Earth. Every scientific experiment we've done on this planet has always had gravity present. And so we don't think of it as a constraint or a variable that can be tuned. We can remove it completely in microgravity, or we can put centrifuges up there, and then we can look at things like lunar gravity or Mars gravity. It helps us understand fundamental physics and biology processes, things that we took for granted. I think it's critical for human space exploration. I think why we're doing this in one part from NASA is to enable further deep exploration of space, astronauts going to the moon and living there and working there, going to Mars. But for me, the point of all of this is, what can we bring back to Earth? What discoveries can we make in space that are going to help humans on Earth? And a lot of these are disease-focused because we've got such interesting physiologic phenomenon that's going on when humans go into microgravity. So I'm just going to give you a few case studies about biology research in space. Of course, we've got to talk about sequencing because that was the most fun experiment. Cell culture and organoids. How do we monitor human health up there? I want to talk a little bit about sustainability and water purification, and then things like synthetic biology and biomanufacturing. So DNA sequencing in space. Why did we do this in the first place? We decided to do this because we were really interested in microbial and environmental sensing. So previously, if you were going to identify something microbiological in space, you would have to wait until a Soyuz vessel was coming home to the planet. You'd have to swab, streak it on plates, wrap it in parafilm, send it on a Soyuz vessel that's going to go through a plasma fireball and hit the Earth in Kazakhstan. And then NASA would send a plane out to pick it up, fly it back to the lab at Johnson Space Center, and only then could your classical clinical microbiology start. It's sometimes 3 or 4 months in between space station flights that are deorbiting to the Earth. So we all know that biology does not operate on a 3 to 4 month time scale. Anybody's come back to see their E. coli culture just absolutely explode after one night going out of the lab. So this was really not very helpful for understanding if an astronaut had a bacterial infection, if we had environmental concern. We needed to know right away. We need to do this in a point of care way. We also are interested in profiling human physiologic changes. We want this to be a tool on the space station to monitor human health and disease. And as we get increasingly deeper into space, we're not going to have these resupply missions. We can't send parafilm-wrapped plates back to Earth from Mars. Really long term, I think this could be a very cool thing for astrobiology and looking at non-canonical bases or things that are not based around our types of DNA structure. So microbes were our first reason to do this, and so I did a whole lot of swabbing on the space station, and it's really fun because you start your experiment on the ceiling, kind of hanging by your feet like a monkey, and swab those surfaces. And so we got a bunch of swabs, and we're taking a look at those, and we're sending those back to Earth as well. And then we decided, we're going to actually do this. We're going to try this in space. But we have a few challenges, right? So we're designing this hardware. We've got to think about power, mass, and volume to be able to launch it. Liquids in microgravity, I'll talk a little bit about that. And then just how are we going to set up a laboratory? I did not have a fully outfitted wet lab. I had a table and a place that I could plug something in with a very specially designed connector. And so if you look at this standard benchtop sequencer that we all know and love, there's no way that something that's this big is going to, number one, be small enough to fit in those little crammed spacecraft. And number two, these things have lasers and CCD cameras, and you jiggle those around on top of a ballistic missile, it's probably not going to work very well for you at the end when you want to use it. And so really the advent of the nanopore sequencer, and if anybody's not done nanopore, it's about the size of half your cell phone. You can stick it in your pocket. It's pretty rugged and robust. We decided, the time is right. This is the kind of technology that you can take off the planet. We also had to think about liquids in space. And if you haven't spent a lot of time looking at liquids in space videos, they behave incredibly strangely. And everybody thinks, oh, well, everything floats in space. It doesn't really. Everything is dominated by the forces of adhesion and surface tension. So if you have some liquid that you squeeze out of a drink bag, whatever it is sticking to, it loves sticking to it. It does not want to move anywhere. So this could cause problems, right? If you are putting liquid in a tube and you get some air bubbles in there, those air bubbles are going to stay in that liquid, and that liquid is going to adhere to the tube. We also have issues with, we didn't really know, how is that liquid going to behave in a container like a 1.5 mil Eppendorf or, strip 8 PCR tubes? How is that actually going to work? We hadn't really done molecular biology before. We also hadn't done much pipetting in space. When I first came to NASA, I, asked about their lab setup, and it turned out we really didn't have one. And I said, well, how are you doing your pipetting experiments? And they said, silly Kate, I was a baby astronaut at that point. Silly Kate, we don't pipette in space. We build an entire box that's a shuttle mid-deck locker, and it handles the fluids for us. And I didn't get it. I was like, I just don't understand why we're not doing this. So when I went up to space the first time, in my support kit, we have a psychological support kit that we can bring with us. And most people put in, cookies and brownies and letters from home and stuffed animals. And I was like, no. I'm packing that thing full of lab equipment. I've got 5 kilos of personal items that I could transport to space. I'd have a little conversation with my psychologist that kind of went like, you know me. At that point, we knew each other really well. What makes me happy? Experiments, research. This is my psychological support kit. It's going to make me happy to send scientific equipment to space. And he got the approvals through. So we really had to understand how liquids were going to behave in space, how we could move them around, how we could pipette them. And I'll show that a little video of this right here. And so these are the items that I brought in my psychological support kit. And we just did this with food coloring. And I just tried moving liquids around. And you can see I've got 50 mil conicals, I've got Eppendorfs, I've got strip 8 tubes. This is a 384 well plate. As it turns out, pipetting works just fine in space. There's a little bit more of a meniscus, and you have to fill the tubes a little less full than you normally would. But I can be a little snarky sometimes. So I just made this video and sent it back to Earth with no commentary. Just, it works, right? It's a little bit more fun because in space, your cell culture plates float, and this just doesn't happen on Earth when you're doing cell culture. But this demonstrated, look, hey, we can do liquid handling in space. We can use all of our tools that we would use in regular gravity. Sometimes we don't have those tools, though. We didn't have a lab setup. But what we did have was a 3D printer. So when I needed to make a centrifuge to spin some Eppendorf tubes, we 3D printed it and put it on a drill. It's a lot of fun doing experiments in space. And a lot of that is because we are out of the box thinking. It forces you to think differently. You are not in a hospital. You don't have a 3-prong power plug. You don't have unlimited data, and you don't have unlimited supplies from a Fisher or a VWR catalog. And so we did this sequencing in space. This worked the first time. I've got the biggest grin on my face because, I'm an okay experimentalist, but stuff doesn't always work in the lab, right? And the very first time we did DNA sequencing, it worked, and it worked beautifully. If you're ever going to have an experiment go right, the first time that you do DNA sequencing in space is a really good one to have go correctly. So we published this back in 2017, and we had sent up a mixture of Lambda, E. coli, and mouse genomes so that we could get a range of GC content. And we wanted it simple enough that if something wasn't working, we could identify it, but complex enough that we could deconvolute this and reassemble the genomes. And from there, we've done a lot more. So we've done RNA sequencing, cDNA sequencing. Then we wanted to move on to this microbiology sampling. And so we did a swab on a wall, we put it on a culture plate, and then sequenced it from that swab instead of sending up a library. So we did all the library prep on board. Then we wanted to do culture independent. So we did surface swabs and we did water that we filtered down and didn't even put them through a stage where we're isolating bacteria and growing it up on a plate and sequenced that. And then there was a really incredible high school student project that again was using this pretty simple equipment. This was it used a thermal cycler that could be built for less than $100 to do this CRISPR-Cas9 experiment with high school students. Like, think about that kind of level of hardware and biomedical innovation and where you could take this technology if you had that kind of thinking in your design. So I want to switch to cell culture and organoids. We also were interested beyond the DNA sequencing, what's happening at the cellular level. And so here, this was the first time that we did long duration culture on the space station. And so we've done experiments on the space shuttle, but the space shuttle was never up for more than 17 days. So we'd never cultured cells for longer than 17 days in space. And so this was a great collaboration with Joseph Wu's group at Stanford in the cardiovascular lab there. And they took human tissues, they transformed them back into stem cells, and then we split them in half and sent half to the space station and half on Earth. We did a lot of different kinds of imaging, genomics on these cells to try to figure out how things were different in space versus Earth. And then we extended that on my next flight, where we were actually looking at engineered heart tissue. And so this wasn't just cardiomyocytes. This was endothelial cells and fibroblasts in engineered tissue. And we seated that between two poles, so flexible poles. So you could actually see the contractile function, and we could measure this and add this to all of our analysis. So this was pretty incredible. And when I would put these cells under the microscope, I'd call my other crewmates over, and like the cosmonauts would float down from the Russian segment, because you're looking at beating heart tissue on orbit on the space station. It was incredible. So we published this work, and a lot of additional publications are coming out about this as well. We also have a great collaboration with the NIH called Tissue Chips, and this is to expand the organoids. So they've sent a lot of these to Earth. There were 10 different tissue systems. I worked on some bone synovial fluid, kind of a modeling of the knee to look at what was happening in the knee during spaceflight. We've got liver systems, we've got brain organoids. This was so successful that they decided to renew the program and do 10 additional experiments, and a lot of those are flying right now. So I think we're going to get a huge body of literature out of this when we get these results back. I think this is just the beginning for organoids on board the space station. I think that's really a huge area that we can go. So human health in long-duration spaceflight. Again, similar to the lab, we have very little equipment in space for monitoring human health. There's no MRI. There's no X-ray. For a long time, there wasn't even the ability to do a CBC or a diff. If we wanted to do a urinalysis, we'd have to float down to the Russian segment and beg the Russians for a dipstick and their machine to do the urinalysis. So we have done some incredibly creative things with ultrasound and other monitoring technologies. But the humans in space, this is one of our resources. So actually, as an astronaut, I signed up for over 25 different IRB-controlled experiments. So my paperwork was literally a stack like this. But we can learn about things like the neurovestibular effects of going to space. When you come back, you feel incredible vertigo. It is like you've transformed into an 85-year-old who has lost their balance and coordination. You lose proprioception. You really lose the ability to navigate yourself, and you completely change the way you walk. If you watch an astronaut who's come back from space in the first few days, they look like an 85-year-old who has balance and motion coordination issues. We obviously have got cardiovascular issues. There's an interesting New England Journal article that demonstrated stagnant or even reverse jugular blood flow. We found this because we were doing ultrasound and came across a giant clot in one of the astronauts. Completely healthy, no history of family disease. It was spaceflight that was causing this clot. We went back and we looked at other astronauts, and we actually are seeing these clots in other astronauts as well. And so, if you think about the way the body's evolved and we're constantly pumping fluid up because gravity draws it down, you remove that gravity, we're still pumping that fluid up, you get this huge fluid shift headwards. We haven't even started to explore what's happening with cerebrospinal fluid. How's your glial flushing? I worry about these things when I'm in space. We've got bone and muscle degeneration. That's been known for a long time. We have 1,000 times higher carbon dioxide than we do terrestrially, which leads to manifest, behavioral issues, cognitive issues. There's a lot of things that we're finding out with the CO2. Immunology, the immune system is degraded on orbit. We actually have viral reactivation of things like herpes virus. We've got waste and nutrition. Those are huge things to think about that you don't take, you take for granted on the Earth when you've got this sort of open loop system of I go to the grocery store and then it goes in the toilet and I don't see it anymore, right? On the space station, that loop is closed. We got to think about that. And radiation's huge on the way to Mars. So I think the future of human health monitoring in space is going to be, again, small portable hardware, wearable sensors. I think commercial spaceflight is going to push us to have human-centered devices. Right now, if you take anything that NASA has built and try to operate it, you need a PhD just to get through the manual. It's not user-friendly, it's not for the consumer market. But as we go into more commercial spaceflight, we're going to have to approach, designing spaceflight hardware in a whole new way, and I think that's going to be a really good thing. I think there's a lot, and Chris will hopefully talk about this more, digital twins and organoids. I think we can use space as a test bed for these remote biomedical devices. And I'd like the monitoring to be transparent. Right now when we do cognitive testing, we have to sit down at a computer and remember things and do symbol substitutions and type, hit the space bar when a number pops up for psychomotor vigilance. This is, frankly, by the end of a mission, this is a test of how much you want to do this experiment, not how tired you are. And so things like, AI in the background that can understand your voice sentiment analysis when you're talking to the ground. If you have something on your computer, when I got really tired in space, I would start misspelling words in a really strange way that I never do on Earth. And so I think there's a lot of transparent monitoring that we can do to understand the behavior side of this and then link that with all the incredible genomics and physiologic data that we're collecting. I want to talk very shortly about water purification. I talked a little bit about these open loops versus closed loops. So water is one of the examples of a loop that we have really closed. It is 97% closed loop efficient for our water recycling on board the space station. We basically don't ship water to space. We've had water up there for the last 10 years now that is the same water molecules. We generate our oxygen through hydrolysis. We bust apart the H2O, we make oxygen with that. We reclaim our condensate through collecting human urine and then the condensate through the air, and we purify this. And so you can see here, these are huge refrigerator-sized racks. Like, look at that guy. This is our water reclamation system. It's incredible technology, but there's opportunities now that we know how to do this to miniaturize it, to get that down to something smaller and portable. And there's a forcing function for NASA to do that because we want to go to Mars. So we're exploring alternative technologies, things like aquaporin-based filtration, nanoparticle-based fibers. We're looking for new kinds of disinfectants. We're looking at coatings for the water recycling system for the pipes and equipments to prevent biofilms. And I think most interesting, because again, genomics, we're now starting to sample the water system at all different points in these water systems to try to identify what's growing there. We're using metagenomic methods for spacecraft water sequencing. Lastly, I just want to talk about synthetic biology. Again, we're talking about closed loop systems, and we've closed that loop for water, but we probably need to start thinking about closing it for a lot of other systems. On the space station, we have a waste issue. We generate 7,000 pounds of waste and put it in a vehicle and send it back down to the Earth and burn it up in the atmosphere every 3 or 4 months. And we ship another 7,000 pounds up there. this is like number one, bad for your carbon footprint. Number two, not sustainable. And number three, not going to work on the way to Mars. And so there's a big effort to try to figure out what else can we reclaim. We have mass and resupply constructs. We've got this need for these regenerative systems for many other molecules. We also have to contend with things like radiation and microgravity. So closing these other loops got me thinking about carbon. Right now we've got this really cool Sabatier reaction. And so that grabs some CO2 out of the air, and then when we do the hydrolysis, it takes some of that hydrogen left over, and that gives us just a little bit more water. And this was one of the ways that we got that water reclamation, that really hard, when it gets asymptotic, that hard last few percent. But then we dump this CO2, the carbon overboard, the rest of it. And carbon's pretty important for bacteria. It could be a food source for a bioreactor. We're throwing all this perfectly good carbon away. So if we can recapture that carbon and then use that in biomanufacturing and synthetic biology. Again, we also reclaim all that water out of the human urine, and then we throw away that dried brine. Well, that brine has got a lot of important molecules in it. We've got nitrogen and phosphorus, for one. Could you reclaim that nitrogen from the solid brine and use that as fertilizer for plants? So I think this is an area that we have not explored a lot, but we could get an incredible amount of research done here. And if you think about everything that I've suggested, can you picture a small portable device, right? Our waste system is terrible. Why are we doing this way? We're using, the same kind of sewers that they used in the 1900s. So it's time for reimagining our waste streams. And I think space will have a forcing function because we are in a total closed environment that could help develop small portable technology for Earth. So I keep saying every molecule counts on the way to Mars. They're all important. We need to use all of them. We need to build our new environmental control and life support from the ground up. We want to embed sensors in everything. Right now, we only have a total organic and inorganic carbon analyzer in the water system to just tell us, like, hey, there's excess organic carbon there, maybe something's contaminating it. Like, we need sensors, very small portable sensors in every aspect of this system. We can think about our consumables recycling. We throw away so much food packaging on board that contributes to our 7,000 pounds of cargo. What if we made that out of a polymer that could be broken down into a monomer that you could put back in your 3D printer, and then you could make more centrifuge rotor drills? And so none of these are necessarily going to be home runs. Some of this stuff isn't going to work. It's going to be too energy intensive to actually make new materials, but we need to start doing it and figuring out where the low-hanging fruit is. And so I want to bring this back to resource-limited environments. I talked about this at the beginning. We have these hospitals, and we've got these research labs in the US and in Europe, and we just assume that all of these consumable type things are given. We assume power, we assume data, we assume clean water, we assume, Amazon is going to deliver a package to your lab the next morning. But this is not a given in almost all of the rest of the world. And so we cannot assume that we're going to have power, equipment space, cold stowage. And we're not designing our equipment and our hardware for everywhere else that's not a hospital or a lab. So when you design something for the space station, you can design it for other areas on Earth. I used to work a lot in the Democratic Republic of Congo. I worked on Ebola and Mpox. And getting our lab equipment out there reminded me almost exactly of how we get our equipment to space. Even if you look at the Range Rover, about the same size as a space capsule, you have to package it for ruggedness. There's no power grid in the Congo. You have to think about where you're getting your solar power from. There's definitely no cloud. A lot of our biomedical device hardware assumes that immediately that you have a connection to the cloud. It doesn't work if you don't have internet. And so designing these things for an environment like space will allow you to use them anywhere else in the world that's a low resource environment. And so just thinking about these things, rural medicine. I split my time between Houston and Chicago, and in Texas, it is a huge state, and people live very, very far from a hospital. And you've got patients that are driving 2 or 3 hours, sick patients that are driving 2 or 3 hours to go get a test done. Now, not everything you're going to be able to take out of the hospital, like MRI, pretty hard to make that remote portable. But there are a lot of things that we could be doing that we could offer to patients at a remote health clinic, that we could offer in their home, that would really help ease that burden, that transport and logistics burden, particularly for people who cannot afford it. Humanitarian disasters, anywhere that there's a disaster, you have a lot of sick people and you lose your infrastructure. For defense, forward deployed units, if you have soldiers that are really far forward and you need to get medical care to them versus being able to evacuate them back to a hospital. And of course, research and medical care in the developing world. And so I want to end with, this picture is so inspirational to me because this is the Artemis 1 capsule. This is a real picture. It's not a PowerPoint. We do a lot of PowerPoint at NASA, and so it's very gratifying to see spaceflight hardware. And you can see that the moon is closer in this picture than the Earth is. That means we are farther out beyond the moon, and we're seeing the Earth in a distance. And we're going to the moon, and we're going to Mars, and we're going to have to live sustainably in these places, and we're going to have to think about transport and all of this. And so as we're building these tools to send humans deeper and deeper into space, I think we can build tools that are going to allow us to do more on our own home planet in regions that we normally have not done this kind of work before. And so with that, I want to thank the whole team that did the sequencing in space work and the heart cells work as the two big things that I talked about. Lots of other experiments in there. I'm happy to give you references for any of those. And so with that, I'll turn it over for questions.
If you have any questions, come up to this mic. We probably have time for one question. And then during the break.
Perfect.
Thank you for the fascinating presentation. Can you talk more about your plans for the future?
Yeah, well, there's a lot of hardware that I want to build, clearly. I think what we need to do is really start some initiatives around the synthetic biology. We haven't done much biomanufacturing in space. I am working with the DoD on small mobile biomanufacturing devices and getting NASA involved in that. But I think we need to do a lot to understand things like bioreactors in space and in situ resource utilization. I think we really need to expand our medical capabilities. We're definitely going to need to do that for the moon and Mars, where telemedicine is the only thing you have on the way to Mars, and it's a 20-minute delay, and you're going to have to do all your sampling inside the spacecraft. So these days, I'm really interested in things like diagnostics, small portable healthcare, how we look at these systems to make small circular closed-loop bioeconomies. Things that you could have in your house or an office building that are going to recycle waste. What are we going to do when we go to the lunar environment and we need to set up a base there and live permanently? And living off the land with lunar regolith and a little radiation and some solar energy is going to be pretty hard. So that's going to teach us a lot about sustainability and recycling. And I think just pursuing the insights that we get from microgravity research, that's going to continue. We've got things, and all the rest of the speakers will talk about this, but we've got incredible advances in regenerative medicine, in stem cell biology, in understanding cancer cells. And so we're just scratching the surface. People think, we've been in space for a long time. The space station's been up there for more than 20 years. It has, but we are just now able to build the hardware and to do these modularized and miniaturized experiments that we couldn't do before. And so I think we're at the dawn of a new space age where we can do these kinds of molecular and cellular biology experiments. So I'm super excited to see what's next. And with that, I'm going to let the next speakers go so you can all hear about it too. Thank you so much.
That's great. So the next speaker is Dr. Chris Mason. I can talk for about 10 minutes of all his accolades and all the companies he has and all the great things he has. He's a professor at Weill Cornell Medicine. But without further ado, I'm going to bring Dr. Chris Mason up.
Great. All right, great. Okay. Well, thanks, Dr. Shekhar. Thanks, Dr. Beheshti. Thanks for the great introduction, Astronaut Rubens, for setting the stage and some of the experiments I had the pleasure to work with her on. And so I'm going to walk you through, the talk title was Current State of Space Biomedicine, but I'm going to kind of take you through aspects of interplanetary biology, some of the technology and portable medicine tools, some of which you just saw a preview of. But I'll walk you through some of the examples of our lab, kind of the highlights reel of the past, say, 10 years, and then the last half a little bit more into what's coming, some of the future missions, future stations, really extraordinary time coming. So I'll start first with the conflict slide. which has for any company I've advised or helped with or founded, which is long, but is not as long as George Church's. So in particular, George Church actually made this slide because I want to start with something from George. He pointed that basically in about the year 2000, we were hoping that maybe we'd get to a $1,000 human genome, maybe about the year 2040, because it was tracking almost perfectly with Moore's law in terms of how the increased density of transistors on chips. And so we thought, okay, by 2040, it's not so bad. We'll get to this case where we have a $1,000 human genome and embed the basic information of your genetic code into medicine, have precision medicine. So it's like, okay, that's coming along. You know, Sanger sequencing is getting better. But then something, of course, happened in the mid-noughties. I like to call the mid-2000s like not the mid-noughties. Does anyone else do that? Is that just me? I'm trying to like popularize this idea like back in naughty 7 when I was doing this work. It hasn't caught on yet, but if you can say mid-noughties, I would appreciate that. So, but in the mid-noughties, there was a really rapid expansion of all these platforms doing massively parallel sequencing. So suddenly the competition got very tight, the technology got much better, and frankly much more highly parallelized, and the cost dropped orders of magnitude and faster than Moore's law. So actually this chart represents the fastest pace of technological change in human history. So that's pretty extraordinary to look at. And of course, as a geneticist, it's great because I love sequencing, I love DNA, I love RNA. So every time you'd plan an experiment, a few months later, the cost got cut in half and then cut in half. So it was actually the best time to plan experiments was in the mid-noughties. But now it's still good now because we got the $1,000 genome. But it was kind of announced in 2014 that it was getting there, was almost there. And then basically the $100 genome came out, a couple years ago. Ultima Genomics announced this 2 years ago at AGBT. And then just recently this year, they announced the $80 human genome. So at some point, the price of a cocktail in Manhattan will be more than the cost of just sequencing your genome. So it's like, then you'll think, should I be drinking this much? Should I be even having any cocktail? Should I just be sequencing my genome again? You should probably sequence your genome instead of having that other drink probably. But the point is, it's gotten now so cheap that you can sequence anything, anywhere, for any reason, of course, for precision medicine. But for many applications, I'll show you some of them today. And so I like to tell this to medical students and people in lab is that what that means is, every day there's more data generated than any other prior day, and it's getting cheaper and better and faster. So every day is literally the best day to be a geneticist whenever I wake up. It doesn't mean I'll have the best day. I could stub my toe, I could drop soap, I could fall out of the shower sometimes, who knows what happens, but quantifiably every day is the best day to be alive and wake up and be a geneticist, and it's getting better every day. So that's exciting, but when you sequence a human genome, we're all excited about this, of course, for most of the cancer patients at Weill Cornell, they get sequenced. Most of the patients at Pittsburgh, I've heard at UPMC get sequenced now. But is it a full genome? These short reads, which have been the bedrock of a lot of massively parallel sequencing, is still incomplete. It's not a full human genome, which really was only done in 2022. Now, you might think, didn't we have the genome completed in 2001? Wasn't there a press conference and Bill Clinton and there was a signing? Didn't that happen? It did happen, but we hadn't yet actually finished all these other gaps of the genome because you require long reads to get there. And that's one thing that was done. The Telomere Consortium, or T2T, really filled in a lot of these gaps in 2022. And what's exciting, I think, now is that this has become a standard, and it's even something we just published a couple months ago, going across multiple generations. So ideally, we would monitor people, patients, astronauts, anyone using long reads, short reads, multimodal, multi-omic data, and even doing it multi-generationally, I would argue would be even more powerful. So when we think ahead, we think, okay, now we could have these technologies and even better sequencing, better tools, and maybe even get to Mars. But the original ETA, was looking at around 2035, then 37, got the late 2030s. But just recently, in the past year, Elon's been tweeting that maybe we could get there. Hoping that Starship doesn't blow up on the next one, but maybe we could get there. It does have some rapid unscheduled disassembly sometime, which is what happens to me on most Friday nights, actually. Just a rapid unscheduled disassembly, and I collapse on the ground. But in 3 years, though, you could have uncrewed flights. You could maybe in 5 years. That's not that far away, right? So this depends on an exponentially growing flight rate. So when Elon first posted this, people thought, well, is there really exponentially growing flight rate? But indeed, there is. So we did a review of this, Afshin and I, looking at all the different flights over the past, basically since Sputnik in '57. And it's actually a version of this figure also made by Matt McKay, who's in the audience. It is exponential. It's super exponential in terms of launching crews and objects and satellites into space. So this is rapidly expanding, and it does beg this question. Okay, we can get into space, but now that is, can we, endure the trip to Mars, as Afshin mentioned? and what are the hazards? And again, Dr. Rubens just touched on a lot of these, so I'll go through them quickly, but there are many modalities of biology that are stressed, that suffer, that have to endure this change. And originally, the Air Force would test some of this, do early testing, for example, just on like cats. They would answer these age-old questions like, do cats always land on their feet? The answer is no. Mostly no cats were harmed in them making this video, but some of the very early parabolic flights were some of the ways we could begin to test this. But this sensation, I've been in parabolic flights but never been in space, but you do have that moment of this unusual sensation in the body, and this is what the cats are doing, what the astronauts have to do, what we'll have to do in the moon and Mars. And so I think these models, and really any bit of data we can get, are what we're trying to integrate and examine across many missions. So with that, one of the missions the first mission we got to work on, actually is where I first met Dr. Rubens, was in a planning meeting, and she was in the back talking about RNA purification. I was like, who knows guanidinium thiocyanate? It was a very great moment of like, who's calling out this preservative in lab? And so it was Dr. Rubens. But we had the chance with the twin study where Scott Kelly was going up in space for 1 year, and his brother Mark Kelly was staying on the ground, who's now Senator Kelly. So we can begin to look at the differences of Republicans and Democrats potentially in the Senate and see why they vote a certain way, and then CRISPR a Democrat into a Republican or something, or Republican Democrat. We're not doing that study. But in any case, what we did for the study, actually, and I'll do this quickly because it's been published, and some of the quick highlights, is looked at many layers of omics. This was microbiome data, metabolomics, telomeres, cognitive metrics with Matthias Bosner, cytokine levels, and we looked for things that went up or down in concert. And one of the more surprising features was that telomeres got a little longer in space, was work with Susan Bailey by qPCR, you can see there. And we thought, well, that's unusual because it's almost like that's the measure of when often they get shorter as you get older. So if it's getting longer, that means you're almost getting younger in space, and you gained a couple inches. And a lot of astronauts do. So there's these headlines saying Scott Kelly got taller and younger, and he lost weight. So it's kind of the best diet plan ever. But the question was, what was really happening? And we first had just done this with qPCR. So we wanted to, getting back to the long read methods, we did this newer method called EdgeCase, where we could map all the individual fragments to the genome. So it looks like this. So you take all fragments of DNA, align them to the telomere and subtelomeric regions, and then you can see with single nucleotide resolution what's present in these telomeres. And so when you look out, we can look for the density of this TTA-GGG motif, which you can see here in green on nanopore data. And then we also looked at it in green here in PacBio data, so we could pick it up in both methods. And then indeed, we can see that the telomeres on average got longer, but we could see the distribution shifted. So you see, it's not a simple story of just on average it got longer, true, but there's also some shorter telomeres, which we've since discussed and published with Susan. And there's a lot more complexity. Like with most things, it's a distribution. Like most things in biology, the answer is usually it depends, and it's a distribution. The same thing is true with the lengths of all the chromosomes that we've seen for this mission and some subsequent missions. But overall, we like to say it was good news, for instance, the fact that most things recovered. But then we started to work a bit with Scott Smith and with F. Sheen and others to look at, looking at 29 other astronauts to see how did they compare their distribution of responses relative to Scott Kelly in blue. And so you can see there's a broad range. Everyone just responds differently to space or any other stressor. And you can see here this range of response either before, during, or after spaceflight. So this indicates clearly that we need more data, need more missions, need more ways to compare. And so that's what we've been working on really for the past 4 or 5 years, is to improve the protocols, expand out access to some of the data, and get more missions. And this is a Space Omics and Medical Atlas protocol, which includes all of different omics layers that we've been talking about with the twin study, but then expanded to more recent methods, including spatial transcriptomics and even skin biopsies. And yes, the stool and urine we kept as brown and yellow for this figure because we thought that was evocative. Thank you again, Matt, for that. Yes. But anyway, it gets you excited about the data. So the first chance to use this new protocol was with the Inspiration 4 crew, working with the SpaceX medical operations team, and they flew up in space. It was a short mission, a 3-day orbital mission, but they got up, of course, took some selfies because they're in space. But they just had a good time, but then also really collected hundreds of swabs. We did a lot of blood punches and did some skin punches and did the swabbing of the capsule. You can see here the crew getting excited, but they also tested the ultrasound using a butterfly device. So the first test of the Butterfly IQ portable ultrasound device you can see here. And then, of course, also did some other testing experiments and splashed back down, landed just fine. So it was really good to see, and they were very excited. We were nervous. Everyone was nervous. They landed. They were very excited because I think from what I've heard, when you land, it's terrifying, and they didn't die, so they're excited here. I think they didn't die as an all-civilian crew the first time ever going up, and some of them only 5 months prior didn't know they were going into space. So it was a really exciting time for them and for the crew. And then, of course, then we got the HEPA filters, got the swabs out of the capsule, and then we got to work. And so this was several years of processing and analysis and work, and we published this a lot last summer. And this was a large package, which you just heard about earlier. but as a whole nature collection of many hundreds of research teams around the world. So I'm here representing a lot of people's work. It's not just our lab's work and everyone in the group, but also many collaborators. And in particular, this work, one of the highlight papers was to see sort of the molecular and physiological changes of the crew, trying to see at the cellular and molecular, physiological level, what's changed in this crew and what can we learn from this mission and apply it then to future missions. So one of the first things to highlight, as I mentioned, this is working also with a grant with Trish, was looking at the ultrasound device, which actually worked. Again, these are people who are not trained astronauts. They were just civilians and got trained within a couple months to use some hardware and go through protocols, and did a pretty good job on the bladder and internal jugular vein. Not as well on the eye, so that took a little bit. They're going to do some work there, but you can see they were able to actually, get the imaging working decently, we'd say. And then we also got to apply, for this protocol, multi-omics data from single cell data. So in this case, looking at each individual cell type and see which cells are the most sensitive to spaceflight. It was a fundamental question we hadn't been able to ask in previous missions. And here we can see that CD16 monocytes had the greatest degree of absolute change, and also unique genes that were changing in response to spaceflight. We also saw a lot with T cell activation, as we saw with some of the twin study data. So it was one new assay, the multi-ohm, what's called Dogma-seq, and also multi-ohm kits where you can look at chromatin. But we also did a cell-free RNA profile, which you can see here at each time point for the crew members, what proportion of the RNA in your bloodstream is from different cell types. So you can deconvolve what the RNA molecule is expressed from, your native state that it would probably be arising from. And you can see B cells, natural killer cells, it's mostly the hematological system is what you're observing. But we did see this spike in hematopoietic stem cells that popped up, and Kate Jamieson's also seen some of these similar trends in her data. So we think that there's seems to be an activation and sensitivity to the stem cell population that we can pick up in the cell-free RNA profile as well. You might be wondering, did we get longer telomeres? And indeed, we did. You can see on the right, in flight, we did the blood collection. You can see it went up higher again. It was only 3 days, so we were surprised that this actually happened that fast, but you can see it did pick up and recapitulate that result as well. And this kept begging the question, is it the radiation? Is the microgravity? What's driving this phenotype? What is it? And so thinking about the model of what happens when you do have longer telomeres, there is telomerase activity, but there's also a non-coding RNA called TERA, which helps guide RNA polymerase II to actually do this direction of transcription to get it longer. So that, well, we do have RNA-seq data on these samples, and we have the longer DNA-seq, but let's see if we can dig it up in the single-cell data and the RNA-seq. And we also did nanopore direct RNA sequencing. So in this case, we looked at for this motif of that basically uracil, and also the specific, you see the density of this motif in the Inspiration 4 crew the day after landing, and also we saw it in the twin study. You can see that spike of red is this density of that TERA molecule showing up. So we could pick up the actual RNA from direct RNA sequencing showing up, and also in the single-cell RNA-seq. So kind of the likely mechanism of how it's moving, we think, is the TERA and non-coding RNA. And then we also did look at, again, I mentioned direct RNA sequencing to see, what happened the day they came back from space and do these long-read profiles. And here we could see methyl-6-adenine, or M6A. You can see, again, spike in red the day that right after they landed, and that goes away again. So we think there's also an epitranscriptome modification that we're just beginning to understand as well. So, and then for the last highlight of the mission, before I get into some of the future goals, is we also wanted to look at, did the skin microbiomes get similar in space? One of my favorite microbiome studies was one by Jessica Green, where she swabbed roller derby players before and after the roller derby. So any contact sport, you do bump into the skin of your opponents. And of course, there's a bit of a microbiome exchange, because that just happens. If you're in a crowded elevator later today, you might be, oh, it's crowded in the elevator. I recommend you say, hey, are we having a microbiome exchange? And then just see what people's faces are like in the elevator. I think you always learn a lot about who's in your elevator when you say that. Or I'd say I recommend it. So I got interested in the microbiome from studies like this, right at the end of the late mid-noughties and the early 20-teens. But then also at a point where our daughter, this is Maddie, when she was younger, at one point, because she's cute here and loves ice cream cones, she's just got back from a marine biology camp a couple days ago. Her luggage all smelled like an amazing kind of smells I'd never smelled before. It definitely was microbiome driven. But my interest also came from when she was younger. At one point, she did grab a subway pole and was very excited. She's exploring the world with her mouth, as many kids do. As some adults do, but she actually also grabbed the subway pole, licked the subway pole, and then just continued playing in the subway. I don't have the exact moment of when that happened, but it was a moment of pure terror. If any of you are parents in the audience, you just know, like, there's microbes that have moved between the mouth and the pole, and the pole and the mouth, and something has just happened there, and I just want to know what it is. So I go home, and I'm looking in PubMed and Google Scholar to see there's got to be some information about what's on the surface is touched by millions of people every day. There was one study on a bus from Porto, Portugal. There's almost no information. So I thought, well, this is crazy. We should have a microbiome map of the metropolis. And so we just went out and started swabbing all over the microbiome, the metropolome, as it was called by the New Yorker, to see what was there. And we found lots of interesting, thousands of species, a lot of DNA that didn't map to any known reference. And this is now expanded into a large global study called Metasub, which is over 35,000 samples taken over the past 10 years. We just finished this again this past June. And some of the quick highlight work, again, this is a bit of a sidebar in microbiome, but I'll come back to space. But this whole global map now gives us essentially this complete map of about 100 different cities around the world, and found 11,000 new viruses. We published the first version of this in 2021. And I've now built a global KMER index. You can type up any nucleotide that you want and see where does it appear anywhere in the world. And so I'll come back to this at the very end, because this gives a planetary scale map of some of the genetic changes, and you can index any bit of DNA or RNA that you have. So this kind of, I'll come back to this in a second. Okay, so back to the crew. Away from the subways for a second. But the crew, we took 10 different areas of their body. We did also the gluteal crease, which I do recommend for anyone doing new protocols. Collecting stool is the least compliant of any of our protocols, weirdly, but the gluteal crease, 100%. No one minds that. But we wanted to see, okay, what do we see for transmission within or between the crew members? And in this case, we could actually see, this is a circles plot where it shows the density of the line as the number of species. And you can see between and within individual crew members, labeled 1, 2, 3, 4, you can see this moving. You can see the capsule, even the space capsule, is moving microbes between the individuals. The armpit is pretty thick. We think that here it's a bit of a rainforest situation on the armpit, which is exciting. I think, again, you might think I'm just in the shower, brushing your armpit, getting it clean. You should think, oh, my armpit's dark and dirty and dank and this destructive, evil place where microbes sit. But actually, you should look at your armpit as this amazing, euphoric, and enticing place for discovery and exciting microbiology. That's what I recommend. Okay, so what's next? So that's what we've got. That's some of the highlights of the papers we've published. What's coming up next? Some of the work, again, is now online. We have data portals that have put online. There's a space biobank that was launched as well for banking these samples long term, so people can begin to access them. And this is, something we want to build up because, we're clearly, laughably underpowered. Like, we have handfuls of samples. We can't really make big answers, I think, to questions of precision medicine in space till we have probably hundreds or thousands of individuals, much like GWAS studies of the mid-noughties. We need more samples, but something kind of like what the UK Biobank has done, where they're doing hundreds of thousands of patients, and I think, as we just heard Visnu talk about earlier, doing this for, even millions of individuals will really start to give us the statistical power to make true recommendations, and there's other models we can use like iPSCs, which I'll come back to, but this model is extraordinary, and there is a United States version called the All of Us program, although it recently got a cut in funding, so now we joke in lab, we call it the Some of Us program. Which is a little bit dark. Maybe it'll come back. We don't know yet. Of course, everything's in flux right now in academia and broadly the government. But this program is still great. The U.S. version, they're also doing short reads and long reads on most of the genomes. And I signed up, so I put my genome and my blood in there. You can log in and see, dear Christopher, here's your risk profiles and here what we think about what you might like. For example, you can click on predictions. It's like, will I like cilantro, for example, or coriander? And I love cilantro. It's one of my favorite spices that there is on the Holy Grail. I could sleep in a bathtub full of cilantro and I'd be just happy with that. But it thinks that I would dislike cilantro, so it's completely wrong when I log in and I'm just like, oh, well, the All of Us program didn't get my predicted phenotype. It's a cute recreational genomics phenotype. It's just cilantro, but I'm not mad at them because it's wrong. I'm glad it's there because we could build better models, or like polygenic risk scores or polygenic phenotype scores. I'm perfectly fine that the database is wrong today. Just I want to hope that I log in 5 years from now and it says, Chris, we think that you probably love cilantro. At which point I would just throw cilantro in the air like confetti and run around my office and then probably go to a Mexican restaurant. I think is what would happen. But anyway, the point being, we need the data. Without the data and the longitudinal data and the better data, there's no way to build better models. And so being able to share and access the data is key. Okay, so that's kind of a highlight of what's been done. What's been the last 5 to 8 minutes or so. On what's coming up. So there's a lot, actually, as exciting as it is time to be a geneticist, it's arguably even more exciting to think about what's coming up in Leo. You heard about some of the sequencing in space. We've done some of the more recent protocols with Oxford Nanopore, where they have these integrated devices, which we flight certified with SpaceX, and those are, in theory, could go soon to some of the next missions. We're coming back to this idea of microbes transferring between space capsules and the crew members. Dr. Rubens just published this really massive effort of everything in the space station a few months ago. So studies like this, studies like we've done, and studies like this can, in theory, become very routine, or like the metagenomics of the water in the stations, and there's going to be a lot of stations. So there'll be Axiom Station, in theory, 2027, 28, we'll see some of the modules coming up soon. The Axiom, you'll hear from the Axiom 2 astronaut, John Schaffner, a little bit. So Axiom's flying crew members already and planning this space station. The orbital reef station looks a bit like this, and so this will also be a pretty giant station that's in the works. VAST is going to have multiple Haven missions. The first one might fly as soon as next spring, summer, so it's coming very soon. The test ones, and there's also, of course, the Star Lab, which is with Airbus and Voyager, which will have multiple layers and a big habitation deck and this big, central tunnel greenhouse. And so these are all planned for the next 2 to 3 years. It's not that far away of what's coming. And there's also, which might even fly the test of the Dream Chaser might come. We just heard last week that this will be flying some experiments probably in maybe November this year. So all these space stations, new rockets, it's really an extraordinary time to see all this capacity of what's going in space. And that's not 10 years away, this is 2 to 3 years away. So there's still big questions. How much does it cost? Who's going to have access? The goal is hopefully everyone can have access, but it'll be likely a mixture of industry support, companies flying missions, philanthropic support, academic and industry collaboration. So it's very interesting to see what's going to happen, but there's never been more capacity or potential than there would have in the history of humanity for what will be up there. And imagine having 4 space stations orbiting our planet and looking up and seeing them, which would be extraordinary. So that's kind of the hardware that's coming up. But then I want to think about what do you do on these stations? So I want to think about going from monitoring to protecting. So one of the papers that came out in the package was, what are different genes that you could monitor, or even look at modifying for precision healthcare in crewed spaceflight? And so in this case, we looked at a number of genes that have been characterized either for animal models or in human clinical data that could be relevant for either osteoporosis or aging or some of the stress markers. So some of the curation of models and data is looking a bit like this. But we also have been looking with JAXA. This is with Masafumi Maritani, who has a digital twin program, like we heard about a little bit this morning, but doing this for each astronaut. So you'd make iPSCs and have them characterized as they're up, and eventually you can even have the crew member plus all the iPSCs around that person. So you have both the OG model plus the derived models all in orbit, and I would love to do that for every mission. So there's some work with JAXA. There's multiple efforts from our lab and others to make digital twins. This is just kind of a rendering of all the omics, which you heard about this morning. And then also FRAM2 just flew last fall, and so this work with Dr. Joseph Borg, who had 2 different sets of experiments, and we've collaborated with him on several of these data sets, and it's ongoing, so that just finished as well. And we pioneered also with Navil Sangina recently is getting CRISPR screens to work that already are on Earth, but could work also in space. In this case, doing multiplex knockouts of thousands of genes in one assay, so you can learn about what the effect is for these different genes in cell culture systems. So this is also something that you could fly up, and we've flown up and could fly again. You could do whole genome CRISPR screens, epigenetic reprogramming. You can lyophilize FDA-approved drugs and put them on plates and fly those as well. So some of these are experiments that are kind of a catalog of things that are relatively easy to do today because you can buy a lot of these things commercially, and some of these protocols we've now developed and validated. So it's actually, again, very exciting time because it's not hypothetical. These are experiments we have working already for protocols, and you could imagine putting up. And then I want to, and 2 other comments is to think about synthetic biology, as you heard a little bit before, but also engineered systems of what we'd fly up there. So in particular, one of the favorite things I've ever learned about is just the fact that you could actually take CRISPR-type systems, which of course do genome modification, but take a deactivated Cas9, which still has the homing beacon function, but doesn't actually cut the DNA, and bring methylation or histone-modifying enzymes to turn on or off specific genes. This has been some great work by Charles Gershbach and many others who've already published some of this work, but there's now multiple ways to go into cells and kind of turn like a light switch on or off specific targets to actually see if you could activate genes or turn off genes. And we've done this in vitro. You can see here for CRISPR-I, CRISPR-A activating or turning off specific genes. So this is very new work using some of these. These are VPR systems, VP64, which so far we think are the best. But you can see you can actually get dozens of hundreds of fold increase in expression for specific targets, which is extraordinary. When I was a grad student, I thought, what if someday we could just turn on and off genes at whim? And we're already in that era, and it's just extraordinary to think that we've come that far that fast. And this is, in some sense, an old idea. DARPA had a program to think about, could you activate a response element if you're going into a radiological threat zone or a biohazard? Could you prepare for it and tweak the immune system so you didn't have any overreaction, for example? Or could it help with allergic reactions, for example? So that's tweaking existing substrates, but also we like to take lessons from all creatures, like cute water barriers like tardigrades, which can survive even in the vacuum of space and be rehydrated. We've made an engineered cell line in the lab that actually has some of their DSEP protein into human cells, and these are HEX cells, so they're sort of human cells, but they're human enough. You can actually see if you do a rendering, you can see H2AX where double-strand breaks occurred. That's actually where the DSEP protein is moving in the human cells. And these sort of hybrid cells. And with a stable lentiviral transduction of that gene, we can get an 80% reduction in damage as measured by 8-oxyguanosine. which is somewhat dose dependent. So if we go too far, like 16 gray, I don't know if you can see that, that was too much damage. But we do also see it's associated with marks known for regulating these genes. So this is a paper that's in review, but you know, it shows that it's possible to take genes that are effective for radioprotection, potentially from any organism, and help use that for a protective mechanism in a human cell or any cell. So that's in the human cells, and the last couple vignettes are, we also just started a grant looking at regolith and getting lunar regolith to think about, could you engineer that regolith so plants could grow better using the Mojave Desert as a model, but using actual lunar soil and getting modified microbes into the soil to start to grow plants on them. And at some point, we'll have to think about what's in the soil, what microbes are present, could you have various cocktails of those microbes? The answer, I think, is yes, and we're testing these out now. So it's another kind of way to think about some of the space science, space engineering that's ongoing as well. And then to close on a couple final notes, I think there are still samples that are being collected on Mars that at some point will come back. We don't know when. Some of this budget's been cut, so it might be in the mid-2030s. But the samples are there. There's been samples that have been collected, air and surface samples on Mars. At some point, they'll come back. This is you can see the rover when it was being made, the Perseverance rover. And so it's very exciting because you can imagine, this rover's there, went all the way to Mars, collected samples here. It was looking back at Earth. It looks like this if you're looking back at Earth. This is looking longingly back at Earth. So it's a slide from John Charles. And we think, okay, we could bring the samples all the way back from Mars. We could biochemically characterize them, sequence them, see what's there. And we wonder, oh my gosh, what if we found life on Mars? Or did we just find this guy's face microbiome who took the picture by the robot? So you want to be able to characterize it. And actually, we worked a lot with Venkat to sequence the clean room during the construction and have a lot of databases. So what we can do is say any piece of DNA that's been seen on this planet or any planet, you can use the same index I mentioned earlier and see anything that's in the clean room, on Earth or on Mars, and quickly say what planet has this been seen on, and it should pop up. And so that does not exist yet, but all the technology and indexing methods do exist, and that will happen at some point, I think, in the next 5 to 10 years, which is pretty exciting. And it gets us to the point where we can start to see if we're going back and forth between planets, do we have microbes moving between them? Invariably, we will. It's just a question of which ones. This is the best of my scientific rendering, our art skills here. But yes, at some point there will be microbes moving between the planets. And so it gets us to this really exciting world where we'll have interplanetary genomics. We could think about other missions, which you'll hear about shortly. So for example, could we go a 3-planet flyby? Could we fly by Mars twice and then Venus and make it back in 19 months? What was originally published in 1956 by an Italian astronomer, Crocco, the Crocco flyby. The answer is yes, we could do this in theory with a Starship. Now this would take some logistics to get ready for. It's also hot when you get closer to Venus. That's something we have to work on. But these are really exciting missions that we can start to have that are not hypothetical, and we can start planning them now, and you'll hear about more of them today, I know, this afternoon. And I think that it will take a village. It will take a broad team of many experts. So I think one model is like maybe we're all like the Avengers. I don't know which one I would be. I would be like some of the fungi on the ground probably. But are we like the Avengers, all coming together from different teams, different backgrounds, engineers, or is it like Voltron? Does anyone remember Voltron, the robot that came together and got bigger from the different parts? I don't know what the best metaphor is, but it has to be one of these. I think where we all work together, learn from each other across obviously at Pittsburgh here, but across many institutions and around the world as well. And so. I'll close with a quote from Martin Luther King, which is life's most persistent and urgent question, which I think about almost every day is, what are you doing for others is something that he had said, but I actually want to put one addition to it and say, well, what are you doing though for people that you'll never meet? And I think a lot of these missions, a lot of these data sets will be for people that will learn from them that we'll never meet, or that they'll work on data sets from like GeneLab, for example, or OSDR, and play with the data, and they'll benefit from it even though we'll never meet them, or even if it'll be long after we're past. And I think that's something that's wonderful and helpful and inspiring for the next generation. So with that, just past time, so I want to thank everyone in the lab who's made this work possible. Of course, thank the lab over the years. It's an older picture, a more recent photo here. Thank, of course, funding from the NIH, from NASA, a lot of coordination work with SpaceX and other agencies, foundations. And thanks again to everyone who's here, collaborators, friends, colleagues, and happy to take any questions. Oh, one last thing is also shameless book plug as well is out there. The Next 500 Years also just being translated into Arabic by Bader Shira. And the biggest thanks actually to the patients, crews, their families, and all the data, samples, and resources that they donate. Thank you very much.
We have a break coming up, but we could do a question or two.
Does anybody have
a microphone?
How many of you want to go to space? We can start with a question. It's okay. Okay, good. In an elementary school class, everybody puts their hand up. It's one of those. That's something between elementary school. Yes.
Oh yeah, come to the microphone.
Hi, Dr. Mason. Thank you for the wonderful talk. I just want to know what your take is on the DNA damage repair pathways that are being activated in space, and how is the lengthening of telomeres via Terra? How does that help us here on Earth, especially for cancer patients?
Great question. So we do the DNA damage repair pathways is one of the most consistently upregulated gene expression sets we see across multiple missions, and we do see it often coincident with expression. You can irradiate cells on Earth, and you'll get some of the same telomere elongation and DDR, DNA damage repair pathways. So we think that is something that's radiation driven. And when you are radiating, for example, cancer patients, you could imagine if you just want to kill the cancer cells, but you could protect the cells around them, there are lipid nanoparticles. You could imagine trying to do a protective area around them, and just to focus basically the radiation is something we haven't tried yet therapeutically, but it's something that would be an application for either radiation therapy, potentially chemotherapy if you get targeted with which cells are being protected as well.
Until like 10:15 in 8 minutes. But the next speaker is going to be online, so because Vishnu said he missed his flight, but he was coming from England. But let's thank Dr. Mason.
Thank you.
Okay, and then we'll come back in 8 minutes.
Astronaut Shaker.
see if we could hear you. Adele, if you could speak. Oh, we don't hear him yet. Hold on for a second. We don't hear you yet. Try now.
Good morning.
Yes. Okay, good.
Excellent.
And
First
then-
of all, yeah. Should I start?
Yeah, go ahead and then share your screen. I think we need for the
Yeah,
presentation.
please
Okay,
do
thank you.
that.
Go ahead. Thank you.
Yeah. Good morning, everyone. First of all, thank you so much for organizing such event and inviting me as one of the speakers in this very interesting symposium. I was in the airport and we had the last minute cancellation yesterday, and we just they just couldn't reschedule for the same day. So that's the reason I couldn't make it. I'm really sorry about that. So the thing is, today I'll be talking about the human metabologenomics for integration of multi-omics data. Basically, I'll be telling about the generation of multi-omics data and its applications in the future of medicine. And also, I will give you an example how we actually drive a story starting from the data integration till phase 3 study. So the main thing is, now can you see my screen? Yeah. Sorry, can you see my screen? Is it okay? Yes. Now, the main thing is, I want to tell you briefly about systems biology and systems medicine approach, and I will tell you how we are actually using such methodology for understanding the human physiology at health and different disease states. And in this context, as you know, that evaluation actually create enormously complicated organisms. And this random and chaotic process only work in a particular environment. And of course, over the years, this complicated process even getting more complex. And to study and understand this complexity is a real challenging task. And in this context, we use systems biology and systems medicine approach. If you actually look at the Rube Goldberg invention, if you really want to understand the entire system, you really need to know how many components are there. Basically, what is the dynamic of individual components, how they are connected to each other, then we can actually have the global understanding of the system dynamic. In this context, our methodology is very similar in understanding human physiology. So basically, rather than focusing on specific cells or specific tissues, we really look at define the individual cell types and tissues in the human body. And then later, we basically see how they are connected to each other and actually how they are contributing into human physiology by simply generating additional multi-omics data. In this context, we have started to work on the generation of the human protein atlas, where we generated the transcriptomics and proteomics data for specific cell types and tissues in the human body. Eventually, we basically defined the molecular repertoires of the human body, and we basically created these biological understanding platforms for biological understanding of the entire system. And we have repeated the very similar approach for the human pathology atlas as well. And in this context, rather than focusing on the healthy cells and tissues, we mostly focus on the human cancers, and we basically integrated transcriptomics data, proteomics data, and also with the clinical data. And we put actually all this information in an open access domain in the Human Protein Atlas portal. So the main thing is, it is of course interesting to understand the human physiology based on a specific omics data, but we also understood that it's actually quite complicated systems, and we need to generate multi-layered of information for understanding the specific contribution of each cells and tissue into final physiological condition. And in this context, we believe that actually from converting from reactive to proactive medicine for the personalized medicine, we need to generate multi-omics data for detailed characterization of the human physiology. In this context, we generate whole genome sequencing data, epigenomics data, whole blood transcriptomics, and tissue transcriptome and proteome, and also plasma proteome, metabolome, and lipidome, and also oral and gut metagenomics data, and in some cases, skin and vaginal metagenomics data, so we can actually understand the human physiology by simply generating this multi-layered of information. In this context, we generate around 1 billion data point per individual, and this data is actually quite, provide extensive amount of information, not about the metabolic states only, and also about the environmental effect on the human physiology. So the main thing is, of course, it is important to integrate all this information with the data from variables, from data from electronic health records, diet information, exposome information, and the imaging data as well. In this context, it is extremely important to generate these large platforms where we can systematically characterize the health and disease and integrate with all other biological data, and eventually integrate and interpret all this information using systems biology and large language models and generative AI tools, and eventually apply it into longevity applications, in the precision medicine applications, and also precision oncology applications. And the moment that you have such platforms, you can actually reveal the molecular insights, you can design personalized treatment plans, you can actually subtype the disease, understand the treatment response, look at for disease risk prediction, identify new markers and new drug targets, and eventually even drive personalized coaching. And we think that this is a great platform for actually studying so many different applications in Earth and also in space as well. So in this context, in our research group in Sweden, in UK, also at our company at Cesial Longevity and now in Visa Longevity, we are very much interested in actually creation of platforms for integration of clinical, multi-omics, bearable, and the diet information. And in this context, to generate all this information, we collect saliva samples, we collect blood samples, we collect urine samples together with the vaginal and skin swab, and also fecal sample. We basically generate all this information in very systematically. And of course, if you really want to generate this data for the different metabolic states and also at different countries and also different continents, you really need to characterize, you really need to standardize the entire process, starting from the sample collection, the data generation, and also data analysis part. And in this context, we generate platforms for actually handling of entire process. And we use systems biology and AI approach. We basically created a data management platform, which I will briefly mention. And also we created these mobile wellness application platforms where we can actually deliver all this information back to individuals and actually involve the participants of the study in the study, and also interact with the patients and also the clinicians. So in this context, in our lab, we established a multi-omics lab in Istanbul, in Turkey. And as Dr. Vishnu earlier mentioned, now we are very excited that another one will be actually established in US. It will be one of the largest in the world, and we are very excited about that. So I want to briefly tell you how we basically created such a data set, how we collected the samples, how we generated all this data, and actually how we use it for different applications. So in this context, in Turkey, we basically initiated this Anatolian Precision Medicine Initiative, and the idea was to collect around 10,000 individuals with different conditions. And in this context, we collected blood, urine, fecal, and saliva samples, and we generated all the multi-omics data that I briefly mentioned, and eventually we use it for characterizing health and different diseases. Out of 10,000 people, by today, we recruited actually 8,000 subjects, adults, and also 2,000 newborns. And eventually, we created now all these data sets, and we basically define the molecular repertoires of each individual, accounting their health status. And in this context, we also think that this information is extremely important for generation of these reporting systems and also AI-based, multi-omics-based applications, which is very exciting for basically with the availability of such data. And in this context, all this information that we generate has been actually used for the whole body modeling of the specific individuals. And in this context, we can actually define this diet information and account this microbiome information, and eventually characterize the whole body physiology and actually study all these physiological changes in response to any kind of diseases and also even any kind of diet intervention. So the idea is the moment that we generate all this information, we basically use the biological networks, and we basically strategies for treatment of such diseases. So in this context, of course, it is extremely important to standardize the pathway, the entire process, as I mentioned. But we basically also generate platforms for handling of such biological data as well. At the end of the day, we generate around 1 billion data point per individual. And it is, of course, challenging to integrate and interpret such biological information. And in this context, we basically created a platform where we can actually handle all this process from starting from the data biological sample collection, data generation of reports, and then delivering all these reports back to the individual. And also, we developed some platforms to share with the community as well, in a way that in the GeneRisk Pro platform, we basically created a longevity-based application based on whole genome sequencing data and identify the potential risk factors. And for the newborn risk, we developed a newborn carrier newborn screening platform where we identified more than 2,000 different diseases, and we actually use all this platform for early diagnosis of newborn of the diseases at newborns. And in addition to that, we also developed an extensive platform for cancer genome profiling and also for studying the status of the cancer in cancer patients. And in this context, we also developed a platform for variant calling information for different type of multi-omics information. And we also eventually developed a mobile application where we can actually maintain this entire process and also deliver all these reports back to individuals in an interactive platform, which I will also tell you more about our mobile app. So the main thing is, of course, the moment that you have such biological information, you have to interpret and integrate around 1 billion data points per individual. And in this context, we actively use AI-based systems, and we developed an AI agent system where we can actually integrate all this information and provide meaningful information back to individual. So the moment that we have such platform, basically individuals can ask their questions and then can actually focus on the specific thing that they are interested. And our platform by today can actually maintain can actually interfere with with individuals' own data and eventually provide even recommendations based on his or her individual or personalized multi-omics data as well. So the main thing is we also think that it is extremely important to keep the people in the process. And in this context, we developed a mobile application. And in the mobile application, specific individual can actually analyze and integrate all his or her own multi-omics data, and he or she can generate actually a multi-omics report. We also integrated the variables information, and by today we have integrated the oral rings and also the smartwatches and also the different type of biological sensors. We also have the nutrient tracking systems, and also for actually following the cognitive process, we also integrated a number of games. And eventually, as I briefly mentioned, there's also an AI integration tool where all this information harmonized and provide recommendations back to individuals. So the main thing is, the moment that you generate such platform, you can actually use it for so many different applications. And in our case, we basically by today started for carrier screening applications for candidate moms and dads, and then for newborn screening applications for the newborns. And also we start using it for studying the child and also adult with diseases. And also now we have applications for longevity-based applications as well. And of course, this also provides extensive amount of opportunities in the precision medicine and precision oncology space. And we think that actually such platforms can actually be extremely helpful in accelerating the drug development and also improving diagnostic tools. And we are actually, after running these pilot programs in Turkey with almost 10,000 people, and together with Phenom Health Initiative in US, together with Professor Larry Hood, with almost 5,000 people, we decided to actually initiate this global 1 million Phenom initiative. Simply, we think that this will actually transform the healthcare industry and the drug development space. And we are very proud that actually this has been launched at the Symposium on Multi-omics and AI-driven P4 medicine at the University of Pittsburgh on May 1st during this year. And by today, we are now completing the establishment of the labs in different locations, and hopefully, our lab in US will be, ready to actually process the samples around August. And our lab in Turkey is actually currently running, and another lab in South Korea will be established around August again, and we will actually start recruiting all these individuals. And we are, of course, very much excited also to expand this initiative to India and also other countries during, early 2026 and also during 2026 as well. So the main thing is, after telling you about the platforms and about our initiatives that we are running, I want to give you an example of how we can actually use this multi-omics data for studying metabolic diseases and how we actually run successful phase 2 studies and now initiated a phase 3 study based on that. And the main thing is, I want to focus on the metabolic disease, most defined as the accumulation of the fat in the liver, which is actually formerly named as the non-alcoholic fatty liver disease. And in this context, it is actually defined as the increased fat synthesis, increased fat uptake, decreased fat oxidation, or decreased VLDL secretion. The reason of such disease is actually a combination of such reasons. And in this context, what we did is we basically created a large biological network for basically integrating all this biological process in the liver tissue. We simply created a model of the liver. And also we generated transcriptomics and proteomics data obtained from the patients with fatty liver disease, different varying liver fat, and eventually integrate all this information using the biological networks that we generated. And in our study, we found that there is a deficiency around serine metabolism, and we further investigated that. It's actually deficient because of the depletion in the de novo serine synthesis. And it was quite interesting to find that even though serine is actually a non-essential amino acid, we found that it is not really present, or it's not enough to continue its functions in patients with fatty liver disease, and also in the severe form of the disease, which is actually called NASH. So in order to understand its mechanism, we designed another study with 86 different subjects, and we simply generated the VLDL kinetic data, and also we generated metabolomics and lipidomics data for these individuals. And eventually, this allowed us to study the liver tissue dynamic, accounting its interactions with other metabolically active tissues, including brain, adipose, and muscle tissues. And after predicting the intracellular fluxes, basically changes in the liver tissue, we found that there is actually a mechanism of there's a pathway. There are a number of pathways which are actually deactivated in response to accumulation of the fat in the liver. And in this context, we come up with a strategy by simply combining 4 different metabolites and actually by preparing a formulation. And eventually we activated the beta oxidation, we activated the uptake of the fat, beta oxidation, and also the GSH synthesis in the liver tissue. And based on that, we basically run a number of animal studies and also a number of clinical studies. After coming up with such formulation, we first tested in mice, and it was very interesting to see that within 2 weeks, we can actually decrease the amount of the fat in the liver more than 50%, which was an actually outstanding results compared to all other pharmacological compounds that we have actually tested. And in this context, we also run a number of different rat studies. And again, it was very interesting to see that the combination that we have is actually quite useful in basically treating treating the liver fat in these animals. And later, we basically run the GLP-like tox studies, then GLP tox studies, and also run a number of phase 1 studies and a number of phase 2 studies. And eventually, we found that actually this combination is actually quite useful in activating these multiple pathways in different tissues. And eventually, it can even be used for treating such diseases, which I will give you some examples. So the main thing is, before we test our formulation in patients with fatty liver disease, so basically, we first wanted to perform a multi-omics study, so we can actually characterize the baseline status of these individuals. And in this context, we basically generated clinical and physiological variables in 56 patients with varying degree of liver fat. We generated plasma metabolome, proteome, oral and gut metagenomics data. And here you see that in different patient groups, you see the differences in the liver fat. You see the differences in the clinical parameters in the ALT, AST, uric acid, GGT, and also albumin and creatinine in creatinine levels. And we also generated multi-omics data for all these individuals. And here you see the differences in the clinical variables in response to liver fat. You see the differences in the inflammatory protein markers, plasma metabolome, and also oral and gut metagenomics data. So it was very interesting to show such differences in a way that different, patients with different, with varying liver fat has actually significant differences in different omics parameters. So what we did is, we really wanted to understand if we need the generation of multi-omics data. And in this context, we basically tried to guess and predict the amount of the liver fat in this patient group based on its specific omics data. And eventually, we wanted to see how the multi-omics data is actually affecting prediction of such phenotype. And in this context, here you see you see that even if you use all the omics parameters in a specific omics, in an individual omics, the predictive score that we get is less than 0.7. So, but instead of using all the omics parameters, simply thousands of parameters, if you actually use 5 parameters from each omics, we got a predictive score of almost, in the testing set 1, and we actually got an independent validation set, which is almost 0.87, 0.89. So in that context, this is clearly showing that why multi-omics data should be generated for characterizing the phenotype of the individuals. And here we also listed the important parameters. And here, as you see, that these parameters are actually mostly known, and also some novel parameters has been also defined. And we actually think that multi-omics data is extremely useful in understanding the physiological state of individuals for characterizing the disease and also characterizing the health status of specific individual as well. So in that sense, we basically designed a phase 2 study using our formulation. And in that, we basically had 30 patients, since it was our first phase 2 study. And this was a placebo-controlled study. And in a way that in 70 days of treatment, we actually found the significant increases in the liver fat in the active group. And also, we have seen the significant differences in the ALT, AST, uric acid, and creatinine. And in this study, we also generated, in addition to clinical and physical parameters, we generated metabolomics, proteomics, and also oral and gut metagenomics data. So we also studied the differences in different omics, and it was actually very interesting to show that multi-omics data is extremely useful in characterizing the health status and also studying the differences in response to a specific, intervention. With that, we have actually applied a very similar methodology for characterizing the Alzheimer's disease patients. And again, we had around 90 patients in this case. We basically look at the differences between the clinical parameters. We look at the differences in the ADAS-Cog. And eventually, we used all these individual omics parameters and also multi-omics data for predicting the ADAS-Cog scores, which is the testing of the cognitive function. And also, we actually identify the key parameters involved in the progression of the Alzheimer's disease. And again, later we run a phase 2 study on Alzheimer's disease patients. And again, we have actually found that there is a significant differences in the severe patients with high ADAS-Cog score in the active group, and we didn't see any differences on the placebo group. And after having such encouraging improvements in the Alzheimer's disease phenotype within almost 60 days of treatment, we decided to run a phase 3 trial, and we basically prepared all the clinical trial requirements, and we will be recruiting our first patient hopefully within July. And we are very excited about that. And in this phase 3 Alzheimer's disease trial, we will be recruiting around 1,000 individuals, and we are expecting that 800 of them will be completing the trial, and hopefully the study will be published once it's actually completed. With that, I would like to acknowledge our collaborators, and of course, I would like to acknowledge Dr. Vishnu, Harsha, and also the entire Peacebook team for actually supporting us for running this all very exciting studies. And with that, I would like to thank our funding bodies, including Newton Alice Wallenberg Foundation, Bill and Melinda Gates Foundation, and a number of EU applications as well, EU funding bodies as well. With that, I would be very happy to take some questions if there is any. Thank you.
Thanks, Adil. We won't take any questions due to logistical issues, but if you email me if you have any questions for Adil, I could always forward that to Adil. But that was a fantastic talk.
Excellent. Thank you so much.
Yeah,
Looking
thank
forward.
you. Yeah. So, all right, and thanks for keeping it on time. That was good. We're back on time. So, I want to introduce Dr. Sarah Seeger. She's a professor and astrophysicist from MIT, and she has a whole accolade, it's like Chris, of many accomplishments and many MacArthur Genius Award and everything else. But you're here to hear her, not me. So, thanks.
Well, now for something a bit different. So 5 years ago, I was part of a team that made a discovery that shook the world of science, planetary science. We detected a sign of life on another planet. It ended up being incredibly controversial. I'll get to that in a moment, but I wanted to say that we, sometimes you just have to go out on a limb to make a discovery, to make change. And to innovate, you have to try new things. So let me take you on a journey of scientific exploration. This story begins actually billions of years ago when a very simple life form, cyanobacteria, did something incredible. No, I don't know about you, but I always think about that bacteria, like that nasty stuff in your fridge or wherever, as not being particularly clever. But this bacteria figured out how to harness energy from the environment, photosynthesis. And in that process, completely re-engineered our atmosphere. And by the way, like poisoned most life forms around it. But what this life did was it had a waste byproduct, oxygen, which we all require to breathe. But oxygen is such a reactive gas, it shouldn't be in our atmosphere at all. Without plants and photosynthetic bacteria, we would basically have no oxygen. So that's pretty awesome, right? Because our life re-engineered our atmosphere, made a gas, and that gas is literally screaming out that there is life on our planet. So I love to think about all the stars out there and all the planets around those stars. And if there's intelligent beings on a planet orbiting a nearby star with the kind of telescopes we're hoping to build, and they look back at Earth, they're going to suspect there's life here. Not from big things like the Great Wall of China or pollution or city lights, but literally from oxygen. This gas that's so reactive it shouldn't be here. And honestly, they're probably fighting over data analysis. They're probably saying there's no way there could be life on Earth because so much oxygen, it's just so flammable. And there's large regional fires. Anyone here like a lab? There's a bunch of lab biologists and chemists, right? I'm like just so confused about how life even originated in water, because most things you have to put like a buffer for proteins to fold. Many biomolecules, they're not even soluble in water. So, they could be out there arguing about us. But what we do know today are thousands of planets orbiting other stars. There are literally trillions in our galaxy alone. And for the first time, we humans, we have the chance to find signs of life on other planets by gases that don't belong. Now, I know you're all like, you want to hear about this. I'm not going to talk about that. That is what has been my main line of research. And I've worked on exoplanets for 3 decades, ever since the field started. When now we know of thousands of planets, we have no solar system copies. We found all kinds of crazy worlds out there, and we do our best to, we're like the opposite of the omics, you guys. You guys have so much data. You have too much data. You're figuring out how to manage all this data. You're going to do like personalized medicine. We have almost no data, and we try to craft like a story out of all that data. But one thing I wanted to tell you, which I think you all are on board with, is that, when I said sometimes you have to go out on a limb, when I started in exoplanets all those years ago, people thought the field would dead end. People didn't, really see the detector revolution coming, even though we have Moore's law, the computational power coming. And now this field is very mainstream, perhaps not here, but in many places all around the world, there's, at least, let's say, 1/3 of all astronomers work on exoplanets. So I have a phrase I say, in the search for signs of life beyond Earth, the line between mainstream, like what is mainstream research and what is crazy, that line is constantly shifting. So now I'm going to go to 5 years ago. I was just innocently with my team working on exoplanets, and we literally tried to do our own kind of omics, not on genetics, but on every possible molecule that could be in gas form at room temperature and pressure. Because although our photosynthetic life gives off oxygen, we're breathing out carbon dioxide, we're giving off tons of hormones, plants, trees, everything. I just wanted a ginormous list of everything that could be a sign of life on another world. We don't know why some gases are signs of life here. And one of the first gases that caught our attention is called phosphine. How many people have heard of phosphine? Like very few. It's so obscure, like you wouldn't have heard of it. It's so toxic to human life. We humans make it as a pesticide to kill things. Bacteria appears to make it, like that thrives in oxygen-free environments. Well, while we were thinking about phosphine as a potential sign of life, it's a great idea for a sign of life on another world because nothing really makes it here. Like it doesn't really come out of anything except humans or bacteria. Well, we were working on this, and across the globe, another astronomer was working on this very obscure gas that even like most chemists aren't familiar with, phosphine. And this other astronomer was working to purposely search for signs of life on Venus. Invited us to join her team. As a result, we made an announcement of a reported detection of tiny amounts of phosphine gas on the planet Venus. We wrote like 100 pages, although admittedly, you'll see like quantity doesn't equal quality, but this was a high quality 100 pages saying that you can't just have phosphine on Venus. Phosphine is pH3. There's very little hydrogen. The energetics don't match up. If you think about volcanoes or lightning or meteoritic delivery, no, you cannot make phosphine even in the tiny amounts found. So this is definitely on the crazy side of search for life elsewhere. And why? Well, you've all learned in school that Venus is so hot. It has a massive carbon dioxide greenhouse atmosphere. It's like so hot at the surface, nothing can survive. It's hot enough to melt lead. But just like here on Earth, if you hike up a mountain or go in an airplane, it gets colder and colder. And that's also true on Venus. And on Venus, up 50 kilometers above the surface, it's the right temperature for life. And although Venus has this massive atmosphere, oddly enough, it's actually the same temperature and pressure as right here in this room. And Venus has clouds. It has permanent clouds that are always there. They're 20 kilometers deep. Just like here on Earth, there's life in our clouds. It may not want to be there. It's getting swept up from the surface and it goes into the clouds. And so that's why we think half a century ago, Carl Sagan first proposed the idea that perhaps there's life floating around in the clouds of Venus. It sounds kind of good. I can see people, yeah, sounds great. The only thing they didn't know back then is what the clouds are made of. They're not made of water. They are made of concentrated sulfuric acid. Now, how many people have used concentrated sulfuric acid? Oh, I'm really surprised. Usually this is like a generational test because in the old days, remember, there were like no car seats and there's like no safety. But I see I'm really curious. I'm going to ask you later what the younger people use it for. It's so awful, guys. It is so destructive. It destroys all of our life. None of our life can survive. And here I'm showing you that's a person with sulfuric acid in that red bottle, pouring it onto sugar, like just the sugar you'd have in your house. And our DNA molecule, remember, it has a sugar phosphate backbone. So this is supposed to be like a proxy for our life in sulfuric acid. Now, one thing about this person pouring this, see those gloves? I'm pretty sure are not acid resistant. Well, look what happens just after a minute, depending on the amounts involved. Look at that. That literally is what happens. I've done that. If you come to visit me at MIT, I will show you that experiment. It is, wow. The sugar just gets dehydrated. You can see how hot it is. There's water and sulfur dioxide coming off, and it literally just leaves this black carbon. We do think this is responsible for why planetary scientists just have assumed for decades that there could never, ever, ever be life in the Venus atmosphere. It is really remarkable, really, that when I started working on Venus and trying to think about could there be life there, what you come across is literally people thinking that sulfuric acid is sterile to any interesting organic chemistry. Maybe that's fair. People use it to clean electronics, use it to kill stuff. But it really is surprising that, in another field, people already knew you can get rich organic chemistry in sulfuric acid in the oil industry, where they use concentrated sulfuric acid to take crude oil to more sophisticated products. So I have a kind of longer story I could unpack another time, but I got into lab work this way and decided to start testing some biomolecules to see what would happen in concentrated sulfuric acid. And when I thought that because I'm talking to some biomedical people, I should show some organic chemistry. So I picked like the simplest thing to show you, amino acid. We put our 20 biogenic amino acids in concentrated sulfuric acid. With one exception, all are stable. Half got chemically modified. So the backbone of amino acid is stable in sulfuric acid. Why is this so cool? Because you know what? A bunch of amino acids are found in meteorites, and meteorites are delivered to Venus. And this is just to show you an NMR, nuclear magnetic resonance spectroscopy. I'm not going to explain what it is, but it's a part per million shift at the bottom, intensity on the y-axis. Your job is to just look at the 3 peaks. Every carbon atom will get a peak here. And these are in 3 different solvents: water, concentrated sulfuric acid, and a mixture. And because all those peaks are overlapping, like you only see 3, you don't, it's the red dotted line, blue and white. Nothing changed. That's our evidence that nothing changed, that we did the NMR and with the amino acid and the different, nothing changed. So it is stable. And I wouldn't need to go through all of them, but just anyone who knows what amino acids are, you can take a glance. And this just kind of shows you in blue, the blue is how it gets modified, how they're getting modified. So our amino acids are stable. And we started going through like a longer list of things. Although our DNA is unstable, you know what? Parts of our DNA are stable. Our nucleic acid bases, guanine, cytosine, adenine, thymine, the latter part of the DNA. And we did a whole bunch of molecules. Now there's this like growing number of molecules that are stable in concentrated sulfuric acid. What I'm aiming to do is to create a DNA-like molecule, not our DNA, but swapping out parts, to show that there is an informational polymer that can be stable in concentrated sulfuric acid. Because I want to come to you and have you believe, not that there is life on Venus, but that it's worth going to search for life, that there's nothing to preclude life. And I know our astronaut mentioned the alternate bases. One thing, little tidbit for anyone who wants a detail is, you know acid, in high school you learn acid wants to donate a proton. Everything's getting protonated. And so that might interfere with the hydrogen bonding that holds the DNA together. So we ordered every alternate DNA base, because there's alternate DNA bases that don't stack together with hydrogen bonds, but with other things like van der Waals forces. We ordered them all and are testing all of those. We work with a drug company, actually, to synthesize molecules for us. And we're working on this. We're getting closer. We literally have a molecule we found. It already exists. How many of you have heard of peptide nucleic acid? Some people have heard of it. It's stable in concentrated sulfuric acid. You know, the glycine backbone, stable. So we are definitely making progress here. This work started out to motivate missions to Venus, but it has taken an entire life of its own. And I'm a proud laboratory organic chemist now with my own lab. So one more thing I forgot to mention. You know, people trying to understand the origin of life in water, they take lipids, amphiphilic lipids, and the polar head of these simple lipids points outwards and makes like a tiny vesicle. We're not going to call it a primitive cell membrane, but we kind of want to just say that to make it easier to understand. Well, we put these little tiny lipids in sulfuric acid, and here's a confocal microscopy image showing you that they're making these little tiny vesicles that actually break off into more complex structures. And sometimes I like to think, like, were my 2 colleagues who did this project so clever because the first thing they picked worked? Or does that mean that there's a vast number of these simple lipids with polar heads that will do this? Actually, you know what? You know how rarely do you do something brand new? It was already in the literature from a few decades ago. There was this like heyday of sulfuric acid when people just studied it for the sake of, and people had been putting lipids in it. Someone in the 1920s even put insulin in sulfuric acid. And they didn't have NMR or mass spec. Took it back out, put it in a diabetic mouse, and the mouse was okay, thus showing that insulin could survive in sulfuric acid. So I just gave you like 5 years and 5 minutes, but we're literally going down that crazy side of the line to go out on a limb and say sulfuric acid itself could be a potential solvent for life. And we're working that. And interestingly, anyway, I could tell you way more about this. There's sort of a growing number of research. We're doing theoretical chemistry. We're trying to, So what is our next step here? And I'm going to get to why I'm here today. But what is our next step? Well, we're going to Venus. I started a consortium I call Morningstar Missions to Venus. We used to call ourselves the Venus Lifefinder Missions. But believe it or not, there was so much pushback. not just from people at NASA. This is not a NASA mission. It's too crazy for large government agencies, but even from our private, current and future private donors didn't like it. So we rebranded, and these are a series of focused small missions to private-public partnerships. One thing we're doing I think should interest you is we're not wanting to be like 4 decades until the next mission. Like our last mission into the atmosphere of Venus was 4 decades ago, although orbiters have been since. We're trying to have a new way of doing space science where we have small focused missions that go repeatedly. That's like building off the commercial space now. That, you know, but except for space science missions. So we're trying to pull that off. It's the first of its kind. And within this umbrella of the Morningstar missions, our first mission is, we just call it the Rocket Lab mission to Venus. We're working with Rocket Lab. If you haven't heard of Rocket Lab, you should. If you get on their email list, they're launching like almost weekly now, the Electron, a small rocket. And what you're seeing there in the top left, that is literally our first mission to Venus. It is our real probe flight hardware. It has, in that picture, just been coated with a heat shield. I want you to think of it like throwing a rock at Venus, only the rock is that very expensive probe. And it has a heat shield, so it comes cruising through the atmosphere. This is a small one. It's going to slow down by its own drag. It doesn't have a parachute. And we have a single 1 kilogram instrument shown with the person at the blue glove there. And this instrument is going to shine an ultraviolet laser through a window, and it's going to look for autofluorescence. And if we see fluorescence, it will signify it's not pure concentrated sulfuric acid, but may indeed have some interesting organic compounds on the inside. We're also going to measure backscattered polarization. I can answer more space questions later. But there on the bottom is we went to Hawaii to test our instrument on the outskirts of the VOG. VOG is like volcanic fog, because you know how Kilauea has been erupting. And we weren't obviously allowed to fly over the volcano. But we did put it on a drone, and it's showing you on the bottom right what the instrument looks like. And there's the drone. We flew it around to test it out. But what we're trying to do is send a series of more complex missions. We want to send a mass spectrometer to Venus. The goal is to find very complex molecules, complex organic molecules, so complex that we all agree they have to be made by life, not just through other processes. Like other would be like the meteorites delivering organic molecules that get built upon themselves to something more complex. So are you still all with me at this point? Because I'm now going to transition to what I thought would interest you most. And it's a brand new idea that's still getting formulated. So it's sort of right on what we'll call the cutting edge. And I like this example because I honestly think it would be perfect for a place like here to collaborate on. So old-fashioned is you take a mass spectrometer, you suck everything in, you see a ton of lines, and then you have to, like a crime scene, figure out what happened. We want a very new way of doing space science. And this new way would involve a simple mission still, but sending a parachute probe to Venus that would go to Venus. So we'd have our entry vehicle. You have a little tiny parachute that pops off the top and bottom, and then inside is your actual parachute and probe. And what I'm imagining is having 10,000 mini molecular sensors. Each of these sensors is targeted for a specific class of molecules, let's say. And so, for example, it could be amino acids or nucleic acid bases. It probably has to be more tailored than that. PAHs, we have a whole list of molecules we're interested in. And what we would imagine doing is literally on this probe have, let's say, 30 real estate patches. Each real estate patch would have to have 300 miniature sensors. And let's imagine we're all here a team, all of us in this room, and you're going to get to pick your 10 favorite molecules. You get 10, you get 10. I'll probably get 50. Okay, I'm the boss. But we're going to have like 100 of each. Now, as this probe is descending, because the temperature and the concentration of sulfuric acid changes, we've released a cover, so each patch gets to do what it's, gets its time. And each of these miniature molecular sensors will send like a no, I haven't found my piece, no, I haven't found my piece, say once a second. And then yes, when it does meet its target, it sends a yes. So this summer, we're doing this 10,000, the sensors don't exist, and I'm gonna tell you what they are in a minute. They don't exist yet, but we're doing an electronics problem this summer, literally an undergrad problem, of how do you keep track of everyone? Like how does each sensor get kept track of and communicate back? So that's happening, the 10,000 sensor project. It's mostly in the computer, but we'll build like one of these real estate 300 pieces. Another student is building a 30 sensor unit that we're going to, so if you're in engineering, you'll love this. We're gonna drop it literally. You can have a weather balloon that you can send to 40,000 feet, literally, just have to let the FAA know. You don't even need a permit. Yeah. And then you burn a wire, and as long as your whole thing's under 6 kilograms, you drop it off. And people send up like a styrofoam cooler. But in our cooler, we're putting 30 sensors just to try this. And we chose these humidity sensors where we'll program them to be yes, no. Like, is the humidity above 10% or 20 or 30 for each sensor? So we have those 2 projects going on this summer. And then we're simultaneously trying to develop the miniature molecular sensors, which we've chosen. And this is a project Chris Mason and myself and Daniel Keller are working on. Carbon nanotubes. How many of you have heard of carbon nanotubes? So you have them here. They're being worked on at Pitt. They're being worked on in a bunch of places. What's a carbon nanotube? It's really tiny, made of carbon wrapped up like in a little roll. It reminds me of like, you know, when you buy if you've ever had the wire fence, it looks like that, like just rolled up. But what they do is they put on a defect. So at the top there with the little yellow atoms is a little molecule. You put a defect, like a puzzle piece. And then that carbon nanotube now is designed to interact with a molecule, a target molecule. And so I'm imagining this probe going down and intercepting the sulfuric acid liquid droplets. And when it matches up with the target molecule floating around, it gets a yes. And that's the plan we're working on. Now this one, they've wrapped it in DNA because these carbon nanotubes are not soluble in water or in sulfuric acid for that reason. They're very resistant to sulfuric acid, which is good. And we definitely have to get some work done because the one that many of them now, here's just sort of a flow chart for you. It's not what we're doing, but I wanted to put it here to jog your memory and to show you it's like real thing used in many places. But people are working on this for cancer, actually. So you would imagine putting your blood like in that high throughput screening. And in each of those wells, there's like a carbon nanotube with a defect that's targeting a very specific molecule. You, in this case, are fluorescing it, and you get that double peaked signature. But when it locks onto the right molecule, that fluorescent spectrum shifts a little bit. Because, you know, you've changed the electron configuration when the 2 puzzle pieces have matched up. Now the electrons are doing something different, and it changes the signature. So in this case, in this cancer thing, they have to do a lot of machine learning because, you know, that shift is really subtle. And then eventually you have your detection. On the bottom, I've put a little miniaturized sensor. It wouldn't be like that. We're not going to do we're going to do something much simpler than is shown here and use resistivity. But that's the bigger picture of what we're trying to do. So it matches really nicely with space medicine because you want to imagine in space medicine, you're like in Star Trek where you're going around and measuring people's disease and sensing it very, very simply and easily. And these carbon nanotubes have a whole lot of different applications. Oh, here's a slide that we just like to show if you needed to know some other applications here on Earth, from diagnostics, from drug validation, to show that, you know, it's in some countries you get medication, but actually it's not the actual drug. Someone's trying to scam you. Manufacturing, biodefense. There's a lot of applications for these carbon nanotubes. We're just trying to put things together that already exist in a new way to work in space and to work in the Venus environment. So to summarize, Venus is an incredibly mysterious planet. We like as a human society, last went into the atmosphere of Venus nearly 4 decades ago. These sulfuric acid clouds, thought to kill all life, they do kill all Earth life, but they actually can support a rich organic chemistry. And these are based on my team's new laboratory experiments. Very reproducible, by the way. I told you of one space application, just to show you how new thinking and collaborations between biosensors and space may actually lead us to finding the first signs of life on another world. Thank you.
All right, we have time for lots of questions. Come to the microphone. Yeah, come on.
Hi, Dr. Seeger. Thank you for that talk. I'm going to ask a question that's probably out there, but how would your experiments account for non-carbon-based life forms, especially if your molecular sensors are honed in specifically on molecules that are like amino acids that are here on Earth?
Good question. So I'll address the amino acid question first. As you know, our Earth life, we use 20 very specific ones, but the amino acid backbone is the same in every single case. In nature, in meteorites, people have found lots of different amino acids. We could envision probably hundreds of thousands or more amino acids. So we'd have to look for something general, you know, like we'd want to have that backbone in mind when we're designing our sensor. In terms of non-carbon based life, that's way out there. People love science fiction to think of silicon based life. It's really not what we're able to do right now, you know, and for silicon based life, by the way, no. If you think about silicon on Earth, do you know where a lot of our silicon is? It's SiO locked into rocks. Have you ever eaten a rock? No, because it's like super glued together. So the whole silicon-based life, it's a bit of an out there sci-fi thing. Oddly enough, if we imagine silicon-based life, it might have a bit more silicon than us, but not composed of that. So I do hate to disappoint, but in astronomy and the search for signs of life beyond Earth, it's really more about what life does. It's making complex molecules, it's using complex molecules, rather than exactly what it is. But we can still fast forward 20, 30, 40 years, and we want to do sample return. And if we can do sample return from Venus, from Mars, then we have more at our disposal to look for unique chemistries. I'll repeat it. I can repeat it, just shout it out. Or...
Great talk. I really enjoyed it. So perhaps a little bit different question. I was wondering how would you define the life outside Earth? Like, would that be similar to what we have here, or like what type of parameters we should use to?
It's such a great question. How do we define life? You know, we have trouble defining life on Earth. And as humans, we think we're so smart, but we're so limited, and we're struggling to answer that question. We have no good answer right now. Right now, we're looking for molecules, organic molecules that are very complex, that just should not exist in nature. But where we draw that line between like molecules that could exist versus ones that are only made by life, that is still definitely a subjective question.
Speaker is Dr. Sylvain Coste. He used to be at NASA Ames Research Center, and he's the one, I've known him for many years. But now he's joining the Pitt Visi Longevity Lab, so he's going to be talking about organoids and digital twins, and he's an AI expert, a lot of computational background. So take it away.
Well, thank you. So actually, I'm going to talk on my own behalf today. I'm still at NASA, basically, I've been spending, I'm off duty since March, and so I've been really spending this time to see, you know, where we are and where we should go and how we're going to do it. So it's been really a great opportunity for me to catch my breath after, you know, close to 9 years at NASA building big data. So at NASA, I was the science data officer for space bio and physical sciences. I was also the lead for AI. For life in space, and I had a lab called the Radiation Biophysics Lab. So I'm going to try to walk you through where I think we are and what we should be doing in the context of space, but also in the context of, you know, challenges that Visi Inc. and other group that we heard this morning are focusing on. I'm starting with this slide. It's actually a very popular slide that my dear friend Ashin has built for the cell package that we did in 2020. I will talk a bit about this working group that built this kind of knowledge, but it's an interesting one. It's showing you basically the, is there a pointer here?
The arrow, the big, there you go.
Oh. So the big arrow is the pointer? Okay.
Is there a pointer?
Oh, I see, okay. So yeah, radiation and gravity are the two big challenges in space, and we heard quite a bit this morning about those two of them, and I'm going to try to focus more on these two questions. In this diagram, you can see that basically radiation is known to induce cancer. It's also known to induce bone loss, but bone loss is also induced by the lack of gravity. So you can see how some of these, what we call space stressors or hazards, can be synergistic in their effect. So cardiovascular issues, immune response are typically affected by radiation, gravity, and also the closed environment. So that's how you read this chart. That's actually very rich. So I'm going to talk a bit about space radiation because that's, as I'm a nuclear engineer from training, and we've been focusing a lot of our time on understanding space radiation. So Earth, the space radiation that we see here are not on Earth, and it's because we have this magnetic field that protects us from those charged particles. And the ISS is also fairly well protected from the magnetic field of the Earth. But there is this kind of interesting cartoon that was done in about 15 years ago, trying to tell the public, the equivalent dose you would receive if you go to Mars, compared to something we understand, X-rays. So a chest X-ray gives you 8 to 50 millirem of dose, and basically, if you were to go to Mars on a 2.5-year mission, you would receive the equivalent of 2,600 X-ray to your chest. I mean, actually, to your entire body. So. Is there any volunteer to go to Mars knowing they're going to receive that kind of dose? If you look at the distribution of basically those space radiation, there's really three that I wanted to emphasize on. The hydrogen, protons, so that's a classic one coming from the sun. Helium is another one, so that's this cartoon here. But then at Z equal 26, you see this peak, iron, and Z equal 26. This is actually, does anyone, basically this is a signature of dying star. When there's a supernova, they release a lot of iron, and so iron is really a signature of dying stars. It's really interesting. And it turns out the work done in Berkeley, where I spent a lot of my time, there was a lot of study on iron because it is the most carcinogenic particle in space. That's the one that will induce up to 40 times more cancer for an equal dose than photons, which is the radiation we know on Earth from the A-bomb data. So I'm going to kind of back up a bit, and we heard about biomanufacturing this morning, but I want to show you something that I think has been extremely successful in the manufacturing in space. It's crystal. And so this is a classic picture from 2014. This actually started at the very beginning of the ISS, and even on the space shuttle, people realized that crystal grow better in space by a big fold. So the crystal at the top, it's Earth. It's actually, that's a 2014 study looking at insulin growth, insulin crystals. And you can see here how they're very small compared to the one you see in space, which are, 0.5 versus 6 cubic millimeter difference. And not only they're bigger, they're purer. And one thing that's actually, so why do we grow crystal in biology? You all know that. It's because we can do X-ray crystallography and detect the structure of a protein, which is critical for quite some applications. But in terms of non-biology, one thing that was done recently is the Flawless Space Fiber mission in 2024. And basically, the fiber optics construction in space has the same advantage. And you see here, fiber optics built in space are much more pure. And actually, last year, between February and March, they were building 7 miles of fiber optics on the space station. So quite a feat. And I would say this could be a very lucrative business for creating much more superior fiber optics than you would get on Earth. So you can really clearly see the big difference here. Now let's talk about space, and we heard the great talk from Kate Rubin this morning on what happened in space. So it's going to be a bit of a repeat, but from a different angle. So this paper is from 2023, and I select this paper because I think it's remarkable. This is actually to scale. This is bone cells that were grown. So we're going to build the complex system of animals and humans from the first basic block, cells. I'm going to step I'm not going to talk about molecular structure. Let's start at the cell level. And you can see here is that those bone cells grown on the space station under 1G, so it's centrifugation versus no centrifugation, you see a drastic difference. This is DAPI, so it's the DNA staining. And you can see how the nucleus in space is much smaller and very condensed. That's really surprising. The other thing you can see here is that the proliferation level Ki67 is very low. So basically there's no proliferation on those microgravity cells versus quite some proliferation with the acceleration. Another thing, lysosomes are distributed very differently, and so autophagy is completely messed up or modified in space. And finally, the fibers you can see here with tubulin, there's those long stretch fibers burst in micro-G that you don't see at 1G. And this is really, to me, if you ask me what I'm looking at here, I'm looking at an aging phenotype. You are basically stopping metabolic activity, you are stopping the proliferation of the cells, you are really into a stage that is, indicating a complete different metabolic situation. And I think it's fascinating because those are cell cultures, so they're in 2D, they're attached to the monolayer, so they tend to be pretty good at proliferating. So the lack of gravity is changing the structure of the cytoplasm, and if you know what's happening with the cytoplasm, it's been shown the cytoplasm is linked to the nucleus and can activate or deactivate gene expression. So it has profound effect, and I think this is the first layer of information. So now we're in space. Why is radiation such a big deal? It's because radiation induces DNA double strand breaks. There's a little cartoon of that. And then typically when you have DNA damage, you have repair and everything's fine. But once in a while you have misrepair or you have unrepair, this can lead to cell death and cancer cells. So it's kind of like the reason why, and we heard this morning, we saw some of this picture, Chris Mason shows is a gamma H2X. My lab, we're using 53BP1 as a marker, P53 binding 1 protein. You can see here, photon exposure 0.1 gray, you see those beautiful dots. Those are actually this DNA damage. If I go to a 2 gray dose, which is a radiotherapeutic dose for cancer, that if you get cancer, you get 2 gray every day for 5 days a week, 5 weeks treatment. That's what it looks like. So you can induce quite a bit of damage now in these cells. And so we have this marker that's really useful to really track the effect of radiation. Now, this is a picture I took at Brookhaven National Lab. So when we do simulation for space radiation, we go to Brookhaven National Lab, and there is a NASA center there called NSRL, so the NASA Space Research Lab. And here you're looking at iron, the famous iron from those dying stars, but they are generated by an accelerator. And you can see the DNA damage along many different cells here. So that's a monolayer of human cells being exposed to space radiation, simulated space radiation. And you can see this trick. Let's zoom more on this. And what you see here is these incredible tracks of these particles. So very different type of damage than the type of damage you see from a photon, the type of radiation we know and understand on Earth. And I spent most of my career focusing on these effects and modeling the effect of radiation. So we can break down the cells, we can actually see the distribution of the energy along this track. And to make a long story short, what you can see here is that along this track, within a radius of 0.5 micron, you have a dose of 26 gray. So 4 gray will kill all of us in this room if we were to receive a whole dose of 4 gray. We had 26 gray, but it's local. So what happens is those particles do not necessarily kill the cell, even though it's a massive dose, because it's very concentrated. It depends what the track traverses. So, and the physics is very different, or the biology, the interpretation of the biology is dangerous, because if you think of it as a dose response, you're going to miss something very important. Let me try to explain this mathematically. Very simple exercise. I'm looking at cells being traversed by radiation, and I'm going to count the number of cells that has 0 traversal, 1 traversal, 2 traversal, 3, and so on. And guess what? I expect mathematically from a random distribution to see a Poisson distribution. So let's see if it happens. And sure enough, we are looking at a Poisson distribution. So what I'm trying to say here is that one thing we say in space biology is there is no such thing as low dose for space. You only have a low fluence, a low flux. And so we often, when we train students, we tell them you need to change your way of thinking when you are in space. You're not thinking of dose anymore. You need to think of fluence, of flux, increased flux. Because whenever you get traversed, the cell sees the exact same damage, the number of tracks. So something very important when we model this response, that's critical. And then we built a bunch of computer models. So that's one of my latest papers last year, where we modeled the DNA damage with this track. We can actually predict the LET dependence, so the energy of the particles have a sweet spot that peak, and that peak indicates where you have the most damage from these particles. So we can model these. We can show the importance of the geometry of the cells in modeling these effects. So now that we've talked about 2D cells, let's move on to organoid sister. So this is work that we've done before I joined NASA in 2011. I was lucky to work with Mina Bissell, who invented organoid. And then she started this work with breast cells. So this is actually a human breast. We start with a single cells, and it grows into a structure that it becomes stable. It's called an acinus. When that structure is built over 14 days, you can actually give hormones to the tissue, and it will generate milk, human milk. So this is a very well-functional gland with one single cell to start with. Breast is, I think, understudied in space, and it's unfortunate because for two reasons. First, we now have a cohort of astronauts that is half female, so we should really think about this. And second, I don't know if you know that, but the only known carcinogen for breast is ionizing radiation, nothing else. Estrogen is a factor, but estrogen is not, this is a natural factor, so that you can't count estrogen as your cancer incidence, but radiation is known to induce breast cancer. And so we built models to, we use agent-based model. I was a big fan of ABM back in 15 years ago, and now I can't wait to play with ABM loaded with AI inside, because this is a very simple model where these ABM had very simple rules about how do I behave with respect to my neighbors, and how do I behave with respect to, oh, did I press something? How do I behave with a cell membrane, and we were able to reproduce perfectly the building of a breast through this model. But this is very simple rules. You can do so much better now with mixing agents and AI together. We heard about the tissue on a chip, so this is the next step. After you have the organoid, we can actually move into this very complex tissue, and we heard Kate Rubin about this, so I'm going to go fast on this, but the Avatar program 2025 this year, BPS, the Biological and Physical Science Division, where I was the science data officer, they are launching this Avatar program, and it's exciting because it's taking us, we can basically create a human avatar of any astronauts by taking their blood cells, turning them into stem cells with iPSC, and then you can derive all kinds of tissue, lung, liver, brain. The power of these techniques really is, to me, the first time I saw this talk work was really on pharmacokinetics, showing that if you put the liver tissue in these chips, you get better prediction of drug effect than if you were to do a monolayer response with simple cell culture. And it makes sense because now you have this systemic aspect. It's not perfect yet, but we are getting closer to a copy, an organic copy of ourselves, which becomes an essential tool for the building of this digital twin, which I'm going towards to. My lab built this brain on a chip. This is the work of Egle Cekanovic-Ute, who was another lead of the radiation biophysics lab, and together we built this brain on a chip, so it's kind of nice. This one is a bit different. We're focusing on high throughputs instead of the avatar. This is very small, but we can put 40 humans in that one plate. We have a blood-brain barrier with endothelial cells and a brain region here. We can show you how nice the brain is. We show that radiation actually messes up the endothelial cells and the basement membrane, the blood-brain barrier. And we show that by running a dye into the vascularization, and we can show that once you have radiation, that endothelial cells blood-brain barrier becomes leaky, which can have a big deal because most of the drug that goes to the brain tend to be stuck at the blood-brain barrier, and so having a leaky blood-brain barrier has profound effect on the cognitive system. We also use this system to look at cytokine response, and so here we show that radiation, 7 days after radiation, we start seeing some very big changes at the cytokine level in the brain using this model. So something that could be eventually flown on the station. That's something I'm very keen on. It's this is actually, I call it the chimeric model. I worked on this when I was in Berkeley, and basically the idea is I would love to see this. So this is kind of like my wish list for things to fly in the space station or on any station, frankly. I call it the promoting system, cancer promotion. So basically this is a breast cells in 3D, it makes this nice acini, and then here you can see cancer cells, they make this blob in 3D, they don't stop growing. Now what we've done back in 2010, I was working on kind of like the perfect ratio between normals and abnormal cells because Mina Bissell had shown that you have the normal cells in the breast can dictate the abnormal cells to stay and behave normally. And there is this kind of like system where you can have a breast that is normal with abnormal cells within it. And then we were showing this in vitro. And basically the idea here is that if we use a system like this, you now have these pre-neoplastic we have these neoplastic cells that are waiting to be promoted and grow out of control. So this would be a great way to test promotion in space. So over the past 10 years, NASA has generated a lot of space biology data, and then I was leading the group that basically built this database. GeneLab was the omics database for everything in space or relevant to space. ALSDA was added later. It's an older database, but we modernized it by using the same structure that we built for GeneLab, and this is everything but omics, so physiological data. And so we have this very massive data set today. We have about 1,000 data studies that you can access and play with. They've been standardized, so I encourage you to go check out this website. And this is the kind of data that we can use for AI and digital twin. One thing I mentioned earlier is like the cell paper I was telling you and the SOMA work that you heard. All these communities were at least started being built in 2018. The idea was to really try to standardize the data in a way that can be reused by everyone. And so we spent quite some time on this. And we started with 20 members in 2018, and today there's more than 1,000 members in these working groups, and they've been extremely productive. And then, one thing we've done is we worked our way through normalizing this data in a way that is more useful, more usable, more visual. We can visualize this data, but we were focusing on just one data set at a time. And then in 2020, 2020, we realized that now we want to bring all this data together, and the normal mathematical formalism doesn't work very well. And so we already have shifted our attention to AI. And so this is the work before we were focusing on AI. This is that cell package in 2020. I really like this paper because we were really, for the first time, trying to bring all this data together. And there was a big discovery that was made. It was really a big deal. We show across the board from cell culture, so I walk you through the cells, the organoid, and the tissue. Across the board, we saw a disruption of the mitochondria, which is very well associated with this aging phenotype that we've been talking about. So I think this is a seminal paper that will stay in it's going to be cited extremely well. Afshin Beheshti was the lead on that one paper. So I'm going to finish on AI, and I want to tell you AI with respect to space. So in space, we have small numbers and we have high feature counts. So how do you deal with this? AI can help us here. We can actually amplify these signals by creating synthetic data. How do you deal with heterogeneous data? Well, it turns out AI is very good at this. We can actually mix different type of data together. And finally, AI can help us transfer the data from animals to humans. So I think AI is going to be a big player in the next 10, 15 years for the field of space biology. And actually, we recognized this in 2021, before ChatGPT was famous. We put a workshop together on AI, and then it took us 2 years to get the consortium, but we brought AI experts, not space biologists. We brought people who were not doing biology. And we took us 2 years to gather the information to create these 2 publications that, I think I would encourage you go read them. They're very nice. One is on biomonitoring and precision health using AI for space, and another one is on self-driving lab, because we recognize that Moon and other places in space will not have always a human to do the experiment for you. So we need to move towards self-sufficient system. And this is the work of many people here. So let's talk about how you build a digital twin. So if you think about life, I just walk you through cell making tissue, and those tissue makes a system which actually release molecules, microbes are interacting with this system, and immunological response is important. And so you can make measurements in vitro data, so you can now build databases that are mixing both in vitro information and in vivo information, so histology, medical imaging, blood work. I'm trying to, let me show you a different way. Basically, you can think of this as an ontology problem. So the human system has many organs. Each organ has cells, and then those cells are bathing into this molecular signaling, The immune cells is that one specific cells that actually walk around this entire structure. Nutrients is important. Microbiome is super important in this entire story. So this is what we're trying to model when we do a digital twin of a human. We have to take into account all these different features. And we heard today the usage of AI agents is going to be very powerful. But let me tell you very quickly a little story about knowledge graph, because this is work we've done with Sergio Baranzini at UCSF, who invented what we call SPOKE, the Scalable Precision Medicine Open Knowledge Engine. And very similar to what I was just showing you, that this is his cartoon, so I took from his slides, and basically showing how the complexity of life starts at the molecular level. It takes you to the cellular level, to the pathway level, the cellular level, and eventually the system level. And to solve this problem, he has used not AI, he has used something else. It's a very elegant way to bring different data together. They're using knowledge graph. And so I'm going to walk you very quickly about this knowledge graph idea. So there's the key paper here, 2023, on SPOKE. So there are databases for genes, there are databases for protein, drugs, disease. And the beauty of a knowledge graph is that you can link these different databases together through their relationship. So for instance, we can link a gene database to a protein database because genes encode proteins. We can link drug to proteins because we know a lot about those drugs and what protein they interact with, how they interact with this. This is no knowledge. You can build these edges across these different databases. Disease, obviously, GWAS is a big one. BRCA1 is known to be linked to breast cancer and so on. So the beauty of knowledge graph is no matter how many data you have EHR, electronic health record, you can bring this in. Because you will be able to find some code that will be linked to some of these properties. And so when we worked with Sergio back in 2021, and I saw his work on SPOKE, I was really fascinated to see what he would do with our data. So we did these quick things where we actually took thymus, liver, and I forgot the third one. Anyway, non-brain related tissue from space station on mice that were flown in space station, and we gave the gene expression and say, okay, project the gene expression values in your knowledge graph and tell me what comes up first. It's a top rank. So what we call, when we talk about ranking here, is like if you do a search on YouTube for something, you are using a knowledge graph. Google has been using this, actually they invented this stuff, and they basically will bring to the top rank, and you can do a top rank based on a given specific thing. So I gave gene expression, but because there was a human database linked to everything, I said, do you have a symptom database in SPOKE? We do. I said, okay, I want to see you top rank symptoms from these liver / thymus data and see what happens. And motion sickness came up as the top rank symptoms for this study. And so to me, this was like kind of an eye opener because it is interesting to see how we could use very simple gene expression data and come out with space motion sickness from a human database using animal data. So I always refer to this paper when people say, oh, animal research is not the best. Like, actually, it's pretty relevant. And this is another paper from Sergio where he basically used this knowledge graph showing that he can actually build signatures. So in his case, after this paper, they actually started inserting EHR into their knowledge graph. So now that information is there. They also have pollution. They have all kinds of different features. They have moved from 30 database to, I think, 60 database. And that's the beauty of knowledge graph. You bring a new database, you don't have to reconstruct everything. You just link your data to the node, and you can keep going. So it's the beauty of it. And in this case, he actually showed that he can build signature for Parkinson's disease that actually, going back to this patient, is able to see the onset of the disease about 8 years before it happens, which is kind of a big deal for PD. So very powerful technique. And I want to finish on, this is my last slide, on something we discussed during, I used to be at the Cancer Moonshot 2.0 with the Biden administration. So the White House put the Cancer Moonshot 2.0, and we're talking about what can we do to bring different data together. And if you use AI, federated learning is a very interesting concept. So how does that work? So this is your typical AI. You have this network, and what is an AI? It's a bunch of coefficients linked together. So it is. It's kind of remarkable how well it works. And so one simple concept is if you don't want to move big data, why don't we actually send to the different data silo the AI? And so you basically send the coefficient to these different places, and you train locally on the data, get your coefficient updated, you bring them back, and we have something called the aggregator that basically brings this data together, averages them, send it back to each data silo until you get convergence to some training. And so we did this exercise on the station last year. So it was really interesting. We use OpenFL for this technique with HP. And basically they have one GPU on the station, and then we also have this, which is here, and then we have much bigger structures on Earth. And what we've done is we use EKG data to show that basically we're able to train simultaneously data on the station with very limited resources and data on Earth with very big resources, and we were able to benefit from both systems and learn and tune with respect to space, but having also access to this huge data on Earth. So there's a preprint here that I put there. It's the work of James Casaletto. This is kind of my last vision for the future for space biology. Is I really, and this is kind of inspired from the cancer moonshot concept. I put this little flag here. Yellow is not too private data. Red is highly private, meaning you're not going to share it. So government astronaut data are in a vault at JSC and extremely hard to reach. But you have other data that are more accessible, and then you have this open access data like GeneLab and simulated microgravity. But you can imagine how AI could be playing a big role here, where we don't have to share or move data. We just send the AI to each of these structures, assuming those structures have to have some kind of standardized structure so that the AI knows how to work with them, because they're not all going to have the same data. So we have to create a master plan of all the standard data first that we have to agree on. And then once you have that system in place, you can really send your AI that will learn from the limited amount of data you have from each of these silos, bring it back, aggregate. And of course, knowledge graph, I think, is going to be playing a big role in this. So this is kind of like my vision for the future. If I were to do this for space biology, that would be probably a good approach. So with this, I'll take any question.
We have time for questions. Come to the mic. Maybe I got a question. So if you had astronaut samples and digital twins, how would you do your organoids, and what would you want to do with that?
Well, so I think we're at a stage where you could really create this very robust signatures from a combination of things. So you would use tissue on a chip like the avatar. So you create this organic twin instead of digital twin. And then you can generate a lot of data from this. Now, you also would use synthetic data. I think this would be, you would benefit from all the knowledge we have in fluidics, in like the organoid, the agent-based model I was showing you. You can generate the stochastic model, and I can generate 10,000 human breasts under different conditions based on mechanistic model, and you can teach that to an AI. So now you can teach the AI with more formal mathematical approaches. So that's another piece of data. And of course, the last one is the actual human itself. And so I think by gathering these three types of data, the synthetic data, the organic data, and the real physiological data, we have an opportunity to build a very complete picture of a person. And then the idea then is we can really predict, where they are and where they're going in terms of their health. So kind of what Kate was mentioning about, all these wearable, this is another thing that would be really helpful on the human side. And the problem is humans, we can't be too invasive, but we can be quite invasive with these other systems. So I think what's coming up, and Chris mentioned this morning, the work with JAXA on the digital twin. I think this is super exciting. They're working on this because that would be exactly the kind of approach.
Follow up, would you be able to apply that to clinical works, bring it back to Earth?
Yeah, so same thing. You could, so for instance, we proposed recently a digital twin of a glioma where we could predict the tumor response during radiotherapy. I think this is actually an easier exercise because it's more contained and it's not a full body that we're building. So I would say, building a digital tumor is much easier than the digital twin of a person. But eventually, and this is kind of what I think VZ Inc. is trying to also build, and we heard this morning is, we need to build digital twin for each one of us. Imagine your phone having your own digital entity. It would be remarkable. And actually, even for a clinical study, once you build this structure, you could actually have an anonymized process where we can share the information. Now, instead of sharing data, you're now sharing an access to one digital entity that can actually be quite secure and still share all this information for building this massive database. So I think we are at the tip of a big pivot in humanity, and I think AI is going to obviously play a big role there. And I hope the scientific community really embraces it and not get scared from it, because we can do amazing things with it.
So thanks. Our next speaker is John Schaffner. He's a commercial astronaut from Axiom 2. Axiom 2, and actually, Pitt, we're finalizing our research agreements of those in MEU, so we're going to have some fun things to do. But we had this morning Kate, but now John's the other angle of it from commercial aspects of it and what he's seen in space from the Axiom 2 mission. And I know Chris has poked and prodded you for samples, so you're going to talk about that.
Thank you, Ashton. And thanks, UPIT, for having me to come and share the day with you. I always feel a bit overwhelmed. Is there a pointer here? Because I know after seeing all these marvelous speakers today that I'm the least qualified to be here, but that has never stopped me. My wife says they know you're not a doctor, right? And they know you're not a biologist, so what are you doing? So I know some good bar jokes and the food's free, so I'll always show up. In fact, everybody comes with these great slides, so I felt compelled to build a slide of my own to show some data. This shows here clearly where all you guys are and where I am in this knowledge base. My wife saw me when I was building this, and she said, "What is that?" And I said, "Well, I'm just trying to get some perspective of where I fit in here." She said, "Well, you got to fix that. It's wrong. You're way too close to all the smart people, and your dot's much, much too large. So, get back and let's fix that." But I wanted to tell you a bit about my journey and how I came to go to space and what interests me and what brought me together in this, and kind of share the understanding of space access, and what it is that we are trying to do, and encourage the momentum to continue to get there. I grew up in the 60s, and I watched, we'll see if this will play if I push something. I grew up in the 60s, and this is what we saw as kids growing up in the 60s. NASA at its origins and at its best, sending Gemini and its missions, the first people to walk in space, the Apollo mission going to orbit. And this was so captivating to me as a young person. It never left. When I looked at this at 10, 12 years old, it was the most fascinating thing ever. And I grew up playing rocket in a trash can. With me and my little sister, she's never forgiven me for this. And my mom never told the neighbors that we were playing in the trash can or why. But this is what our life became, and this is what stuck with me. In this image, America's first walk in space, the first human from the U.S. to walk in space, Colonel Ed White, really, that is the image that I always remember when I think of space. I was 10 years old when I saw this and could not believe that. How does this work? How does this work? And it drove my entire life. And the next year in art class, I painted my own version of that. And I actually took a copy of this to orbit with me when I flew, because this was just a huge impression on me of what space was. And today, young people today are still faced with these great images of what's going on in space. They're seeing things like this, the most unbelievable things that challenge our imagination to actually grasp and understand. But it's a driver. It's a driver that is going to encourage the next generation of people coming up behind you to continue the quest that you're so boldly starting on today. Now, for some reasons that are a mystery to me, I did not follow a path into the astronaut program, even though I was so passionate about it. I spent life as pretty much an adventure athlete, doing things that were core to me, flying planes, cars, trekking, doing lots of things. But I think all of those were really a surrogate. You know, the things that impressed me when I was 10 years old are spoke of who I was, and I was looking out for the things that I intended to do with my life. But something really marvelous happened about 5 years ago. My wife and I were having a morning, having coffee, engaged in nice quiet conversations, looking at our own laptops. And she stopped and she turned her laptop around and said, "Look, you can finally go to space." She turned it around. It was a news article of Axiom Space. The private astronaut program was here, and you can create an astronaut program. And I could not believe it. I was just sitting there thinking my life was over. I would never go to space with all these dreams, but there it was. But Axiom was a young company. The website was new, and there was no phone number. There was nothing. It just said info@axiom.com. So I whipped out this email real quick. I said, "Okay, I'm your next guy. I'm ready to go. What do I have to do?" And I sent this email, and I sat there by the computer waiting. And I waited. Nothing happened. I finally left. I was hungry or something, came back. Nothing all day long. Nothing all week. Finally, I just gave up. But mysteriously, well, as it turns out, nobody checks info@axiom.com, right? info@anything.com is a loser. But I did make a connection, found a flight surgeon just by happenstance, and I got signed on to the program, and we ended up building a marvelous mission with a world-famous, well-known, and a marvelous person at that, Peggy Whitson, and a couple of Saudi astronauts on Saudi Arabia's first inaugural national spaceflight. And we put together AX-2, and when you go to space, you get your own patch to remember it by, so you remember you actually did something. So we were able to develop a really powerful mission for us, and Saudi Arabia had a good time. Axiom continued to learn, and we learned how to integrate into the existing space program at NASA. So we put together this really awesome trip that was packed with great science and student outreach. You ending up on your launch day, packed into your Dragon capsule on top of a Falcon 9, and sitting in the flight deck. I was the pilot along with Commander Whitson. This is a dream come true for a young person that dreamed of space finally taking off. And as Kate mentioned earlier, it takes 9 minutes to get to orbit. I like to think of it as from here clearing the tower at Kennedy Space Center in Cape Canaveral to 200 miles over Ireland in 9 minutes. I can't find my car in a parking lot tomorrow morning in 9 minutes. But technology today and getting to orbit and all the things that get you there to this marvelous place, the International Space Station, was a dream for me. But a lot of really cool things go on there. It's a nice, wholesome flying laboratory that was put together over the course of a number of years. It's been flying for 25 years, fully staffed, with a complement of international crew. It's a marvel of technology, and it's working really, really well. You hear various stories about it, but it's quite a machine. This is a picture. I want you to watch this. This was shot from our approach from the window of the Dragon spacecraft on an iPad. As we're approaching the station, we're catching up to it, and there it is. I love this image. It's like coming home to something. And I know Kate will probably like seeing that. It just is a thing. When you catch your first glimpse of the ISS coming in on its orbit, you're both sailing along at 17,500 miles an hour. It's a really, really cool thing. And you join a marvelous crew. You get up there with the station crew, the permanent crew that's on board. Everyone bonds and develops a really awesome relationship, and it's something that is with you forever. So as a young person, starting off in a trash can, piloting a Dragon space vehicle, going to orbit, and doing this, I don't know what to think of my life after that. So It was a marvelous opportunity. I was very privileged to have the opportunity. But you do work hard, when you set up to do this. There's a lot of training involved. We trained at the Johnson Space Center primarily and SpaceX facilities. This is Building 9, where they have a full mock-up of the International Station, and you go through all facilities. Mainly, as a private astronaut, they want you to know 3 or 4 things about how to feed yourself, how to poop correctly, and how to run from a fire. But I'm kind of nosy. I wanted to learn all the systems, so I got various levels of certification on all systems of the station, and I found it very enjoyable. We also trained on some of the mission objectives. You have to back that up. This is some of the biology training, the sample prep that we were tasked with to go to orbit and to conduct. So you get to do the entire process of preparing for yourself. And in space, on the station, you do handle a lot of unusual materials, things that are caustic or dangerous, and so you have to get specific training in those things. And there is one training aspect that I think most astronauts dread the most, or most concerned about, the things that no one really likes, and that is, of course, toilet training. You have to learn to poop correctly in space and use the toilet. The problem is there's only one of these on the US side. And if you break it, you instantly lose all your friends. You're the guy. It's built by the Russians. It's very robust, and it works pretty well, but it's got a lot of switches and buttons, and you even come up with limericks to remember that everything's in the right spot. But the problem is everything floats in space, and you get one little particle gets away from you, and it gets on your clothes. You're wearing the brown stain of shame all week. So everybody wants to get it done and get it done right. So astronauts are a little concerned about this aspect of it. But other than that, the view from space, as Kate showed you earlier, is a beautiful, beautiful thing. You get to watch the Earth go by on a complete orbit every 92 minutes. You get to see incredible things. Now, if you go outside on a nice day, you look up and you see a beautiful view of the sky. From orbit, you get these amazing views of a sunrise, and it makes clear exactly what our atmosphere consists of. Here you're looking at the entire thickness of the Earth's atmosphere as it exists. It's a very, very thin skin of protection. It's what protects us from all the elements, that and our magnetosphere. But this kind of, once you see this, you get a really, really good example and understanding of where you are in the universe related to the Earth. And some more pictures inside the station. I like this one because it shows the ultimate apparent congestion when really it is not. To the lay eye, you think, how can anything go on here? But it's very well organized. And when you go to any particular station and look, things make sense. You're trained in all these different parts of the station. But it's an awesome, awesome image of looking down the U.S. lab section of the station. And of course, because you're in microgravity, every surface is a work surface. The ceiling and the floor, they're all the same. There's Dr. Whitson down below doing something, and I'm probably trying to find my lost iPhone. I don't know what I'm doing up there, but you can work anywhere, and that's the efficiency of microgravity and the beauty of it. Now, astronauts' time in space is very tightly regulated. This is a view of our working timeline when you're in orbit as any sort of flyer. Your day starts off when you wake up. The time of day is the light blue bar across the top, and these bars, each of the 4 segments, horizontal, is one crew member. So their tasks are tightly outlined and scheduled. So you have, it's not shown here, but there's usually a red vertical line which represents the time of day, and that slowly slides across the screen, and each task comes up in order. And they are typically tied to must be done at that point or expected to be. You can click on one of those sections and you get a display of information that's pertinent to the task, either procedures, items that are important to do, and whatever. But it's a busy, busy day in space. Now, we've handled the how do you poop in space? Like, we get that question a lot. But we also get asked, how do you sleep in space? And when you're a visiting crew member, you're kind of camping out. So the answer is you sleep anywhere you want. You just get your sleeping bag, tie it off, and park yourself into a comfortable position and go to sleep. And when you're asleep in space, you have no idea where you are. You can be vertical, horizontal, or whatever's going on. So we like that. And all those great pictures that you see in space come from the cupola. Kate showed you some great images from there. This is a little picture of how you drop in, and I always love doing this because everything changes when you drop into the cupola. The sound changes, the feeling changes, and of course, you get that marvelous experience of wrapping around through the Terminator, the sun's Terminator, every 45 minutes. So it's a very, very special place. And of course, you get your hero shot, when you're hanging out in the Koopa a little bit. Earth return, finally, when all the fun's over, you get to come back in your Dragon capsule and you're loaded up and you re-enter just like the great meteorites you see that come in. It's actually a lot smoother. I don't know how yours was in the Soyuz, but it's not violent. It's under canopy. It's a little shaky, but we really enjoyed it. But that's what it looks like to re-enter in a Dragon and splash down in the Gulf of Mexico. SpaceX likes to land at night when the ocean is very smooth. They have some limits, wave limits to the Dragon capsule, but that's a freshly returned. We were on the water about 20 minutes at that point, and they're there ready and waiting. And here you go, coming up out of the water, a fresh re-entry from space, and then you have a nice warm astronaut ready to put his feet back on the planet. So that's the whole experience of getting there, and I'm very happy to have had the chance to do it. And hope what this shows you is a little bit about the accessibility of what this is. So I was an ordinary guy doing ordinary things and got an opportunity to go to space. So I went. And we conducted some really strong and important science, we believe, and we had some great success, which I'll share with you in a minute. But it shows you that this is not coming, but it's here. This was 2 years ago, and there have been 2 other missions since. There's an AX-4 is in orbit today with 4 astronauts. So you and your colleagues and people behind you will have these opportunities. So the message is plan for that. Execute your work today as if it were a possibility and that it's going to happen. Because the science that is available to do in low Earth orbit, Kate showed you a lot of the great work she did in her missions, the first sequencing, and there'll be a lot of new firsts coming up. And although NASA may present certain challenges to get things done, either on timeline or an expense basis, what we're expecting is the commercialization will bring about a relaxed environment to work in. They'll still maintain a high level of safety and performance requirements, but the speed to iterate, the cost to get mass to orbit is expected to continue to mitigate itself and to come down and it be very, very accessible numbers. So when you think about this laboratory of what goes on 250 miles above Earth, think about that you are able to put things there, that you're able to make things happen. And there's been some talk, we've all talked a lot today about why microgravity does work or does its magic. And when you think about many of the formulas, the processes, the physical laws that control many of the processes that affect your biochemistry, affect manufacturing, things that flow, move, many of those have a component of gravity. So here are many of the formulas that you're familiar with today, the Rayleigh number, you've got Morton's number, they all have a component of gravity as a functional element of that. So what happens when you remove those? When you move the G out of these formulas, things shift, and they shift unexpectedly. We do know that many of those move in known directions today. We already know some things increase or decrease in values and in effect. Those are certainly well known. We have some physical force measurements that we have made in many cases. Lots of experiments have already demonstrated the results of what these changes have resulted in. And some of the biological systems checks that many of you researchers have already proceeded with have demonstrated that those are true as new laws of things in microgravity. So which is very exciting. It means you have a whole new environment that you can lean against to try new processes. And these processes then will play value to what we can do to improve things on Earth and also learn how to use those to live longer and to proliferate in space in order to thrive, which is going to be important because we're going to Mars. We know that. It's in our nature. In fact, when President Eisenhower formed NASA in 1959, we thought we were doing it. Most people thought, well, we've got to stay ahead of the Russians. They've got this Sputnik thing, and we're under threat. We've got to start an agency to defend ourselves. But his statements were, I signed this document here today, the formation of NASA, not because we fear for anything, but because we are meant to explore. That is in our nature. He recognized that. And that's the watchword that we all go forward with today, that we are in a nature of species of explorers. We want to know what is the next thing, what's over the hill. We have to know that. So I won't dwell much on this because these are things that everyone well knows, the drug discovery opportunities, nanotube performance, things that happen in microgravity. Some great drug events have happened, discoveries based on things like adhesion, things that work better in microgravity or differently, give you key insights to what can happen differently on Earth. They advise us onto the properties that we can look further at here in our own environment. Lots of great papers have already been presented on this, and it's endless. And I expect a sea of these things to continue as we continue to move forward in our involvement in space. As the Spaceomics, as SOMA continues to improve its database, and as we just learned, AI will have more access to more data and become more effective as a tool to make references and give us compounding efforts going forward as we improve our knowledge of how to perform in space and what is important to know. This goes on and on. So as we do develop more access to space, these platforms become available. The ISS was just the beginning for us. It was the thing that proved that it can work, and it did it so very, very well, and it's still doing it very well. But we have other things that are coming up. Chris has shared with us many of those already. We know that Axiom, working very hard to get their first module in space, so we expect to see that up by 2027, late 2026 perhaps. You have Orbital Reef, Starlab, Airbus with their grand plans, and they have a pretty awesome structure planned. We hope that works out for them and for us. And of course, VAST with their plans. They'll have their center module. They're trying to be the first in orbit, so it's kind of a horse race right now, who wants to get there. But it doesn't matter. It doesn't matter. In 5 years or 10 years, we'll have multiple platforms, and there'll be lots of opportunities for researchers like you to access space and to get the things done that you want to do. All it means is your cost is coming down, and that's the cool part. It's more accessible. The ease, the speed of getting it done, the iteration rate will go up, and that is what's most important to you. So yeah, these things will be pretty cool. They're things of science fiction. Now I came back pretty jazzed from going to space. You know, I didn't expect it would happen in my lifetime, but it did, and I tried to make the most of it. So when I came back, I had all this experience and working knowledge. I wanted to put it to use, so I started a foundation called the Perside Foundation, meant to drive education, because I got my start when I was 10 years old because I saw these marvelous things and I wanted to be a part of it. And I know that there are 10-year-olds out there now that see the same thing. They're looking at a Starship booster come back and go, wow, how am I a part of that? And I grew up in a very poor part of the country in Appalachia in southeast Kentucky. The schools are very poor. and they are still very poor. The teachers struggle just to teach basic materials. The states require basic learning levels. No one knows about this stuff to an extent that a teacher knows how to build that into a curriculum. So I wanted to help change that. So in the past 2 years, I've visited dozens and dozens of schools and classrooms. We have sent teachers out to learn how to become better educators with project-based learning, learning about space initiatives at NASA. It's been very thrilling. And because I was kind of a quasi-artist, because I like putting things on paper, we started the Space Art and Poetry Contest 2 years ago, where we asked students between the ages of 5 and 18 in 3 categories the question, what would it look like if we lived in space? We just asked that one question. And over the course of 2 years, we had over 4,000 submissions of art, the most amazing stuff. You should go to our website sometime, Space Art Contest. Just go to the bottom and browse through the endless pictures that these young people have created. And the thing that's so amazing is we know space is not taught in school to a real extent. But they come up with these images on their own. It's in their head. It's innate to them. Somewhere they're absorbing this thought and presence that space is interesting to them, and it draws them, and they continue to produce these drawings. So with cooperation of NASA, we take some to orbit. They did it a second year for us. We print them on the station. We pick some winners. And because it's art or poetry, we pick winners from each category for art and poetry. And we take a nice photograph of them, and we send them to the winners. And they get a piece of their art float in space. So it takes a while to get it done, but it's pretty cool. So I wanted to share a few of them that are here. I thought that was someone likes Donut King, I guess. Not sure what that was. I guess that's our solar system. 7-year-old from Greece. And this one was kind of fun, I thought. You know, surprise. But and some of our other favorites. Okay, that's probably, hopefully it's me someday. And I just thought this one was kind of fun. It's a good one to end with. You know, teenage drivers are always going to learn to drive no matter what planet they live on, right? So they've got to terrorize their dad doing that. And the creme de la creme for me is always when I'm with a classroom and I look at these 7, 8, 10-year-olds, and I just wonder, how many Martian astronauts am I looking at in this room? How many researchers are there? Because that's who's going to Mars. It's not going to be me. It's not going to be likely no one in this room, maybe, but likely, more likely, some of these folks are going there. So putting that image and that thought in their head early is what was key. That's what affected me. And I think everything that I did in my life since that image of Ed White guided me in a general direction into the quadrant that eventually led me to go to space. So bringing that back and returning that's very important to me. I want to end with some great pictures. When we talk about space imagery, my friend and likely Kate's friend, Don Pettit, always takes some incredible photos. So when Don's up there, he cannot help but wow us. He did these some time ago, but Don just came back from space a couple months ago, in fact. Great time-lapse photos from Don Pettit. These are thunderstorms that you see streaking over who knows where, Europe or someplace. The cities are going by and captures thunderstorms. So the wonder of space is in front of us in so many different ways. And I just know all of you are doing your part, not just to create great research to understand all the omics that are involved in the human physiology to live to go to space, but you're helping create a wonder in everyone and letting people see the results and the value of being in space, not just so we can go and live there, but also so that we can help improve things that we want to improve and change here. So, you know, my hat's off to all of you for the hard work that you all do every day. I'm just a participant cruising through, kind of like that that new cosmic object they just spotted this week. Okay, I just flew through this ecosystem. But I'm trying to stick around, grab an orbit somewhere, and do something of value. But I really think it's inspiring the young folks that are coming up behind you, because you're here because something inspired you when you were younger that brought you here. Thank you. So the rest is up to you guys.
Thanks, John. We have time for one question if people want to. Oh, yeah, yes.
So this is probably a very naive question, but does your sense of time change in space? Do you feel days are longer, shorter? Do you feel less tired, more tired after one day's work?
Well, I had an interesting experience along that line. Is space adaptation is a very real thing. I don't know whether it's age related, because I flew as an older person going to space, but my adaptation to feel normal and effective took a long time. It was slow. So the days felt shorter because I never felt like I was on time anywhere. I always felt like I was slammed to get there, and if I got to bed by 10 o'clock, I barely made it. So it was, I hear it gets easier if you're there longer. We were only there 10 days, actually 9, so I had less time to adapt. But I hear different stories of other astronauts taking longer, shorter, but it's this brain fog. They call it space stupid sometimes, but you see I've got some batteries here, I need to replace the batteries. Oh, I need to replace the batteries. I'm going to replace the battery. It can take you twice as long to actually engage and do things. It did for me. So time was an issue.
But last but not least, we're going to have a panel session after this. But Danica Vallone is going to be talking about all the great initiatives she does. She founded OSMED, which she'll talk about, and this new program that she's starting. So
it still in progress? You
Not on
committed,
this, not on this arm. This arm's good.
Okay.
I think in general, there's always more to do.
Yeah. I'm still working on the nerve to get a tattoo. I want a tattoo.
So I want to first introduce Savi Glow, which you haven't seen yet, but she's CEO of BioAstro, which Chris had mentioned briefly is a nonprofit. But at least Savi, you want to introduce yourself first before we...
Sure. We're good? Okay, cool. CEO of BioAstro, we are a nonprofit. We work to close the gap in understanding human health and biology in space with the intention that that knowledge generates technologies that spin out into the commercial marketplace. And on this stage with me, actually there's only two people on this stage who are not in some way involved with BioAstra. Afshin is on our scientific advisory board. Chris is our chairman. John is recently joined at our leadership circle. Sylvan is one of our executives. Kate is one of our advisors, soon to be more. Danica, we can't wait to have you involved, and same with you, Sarah, too. So everybody, that's one of the hallmarks of BioAstra. Everybody is welcome to the table.
So that's great. Let me grab my... I was trying to keep track of time, but I'll grab my phone in a second. But let me start the conversation here. So I'm talking about breakthroughs and optimism, for space biomedicine. So the questions I want you guys to answer is like, what are the most exciting recent advancements you think that have happened? I know you guys presented a lot of the exciting investments. What kind of highlights are specific for, ways we can work closer to like long duration missions to Mars? And then what types of emerging fields like organoids, Sylvan talked about, genomics like Kate and Chris talked about, or things related with, like what Sarah talked about finding future life on Earth. So those are like, what are the most exciting, like, so we could start with you, Sarah, like optimism and the breakthroughs in space. So like
I
that.
think it was really what was covered. It just boils down to nanosensors and big data.
And I'd say, some of them came up, the ability to have many versions of avatars of yourself, or the organoids give you a chance to really increase the sample size. So instead of having just one crew member, you can have thousands or even millions of copies of a crew member and variations of tissues. And it gives you a way to test drugs, to test biology, to understand biology and the perturbations as well. I mentioned briefly, but there's a lot of tools that are also automated that those kinds of experiments, and also long-term cell culture. So I think as Kate mentioned, like cell culture in the shuttle era had never gone more than 17 days. Now you can do months, or maybe you can imagine doing years in flight if you get something that would go. There's plenty of orbital trajectories. You could just launch something that would just stay in orbit for several years. The Roadster is still in orbit, as most people know. It's just like floating around somewhere between Earth and Mars. And they should have put some cells in there. I don't know what else they put in there, but they put a bunch in the car. But you can just plan missions like that now, which is pretty extraordinary.
I'm going to go into the human and say that the new normal humans, I'm going to call them, our everyday humans who are flying in space, as opposed to the NASA superhumans. Mostly just saying super trained versus everyday trained. That's what I find most exciting about where we're at.
Yeah, I think Chris touched on the Moore's law aspect of technology advancements, and I think there's a couple things there that just the pure computing power. When you think of the Voyager spacecraft, it's just about to cross a light day in distance from us. It's still processing. A key fob on any of your cars right now has more computing power than the Voyager spacecraft had. So what we're doing today is such an enabling thing, and what's really kickstarting that, I believe, is its accessibility. You look at the App Store, it's caused people to think about creating things to do things. And that has such a leapfrog effect on people that they can't stop. It's empowering. So you throw technology and you throw accessibility, like a gamified platform, an app, do things in commerce. That's throwing gasoline on technology that's just taking off. So that's going to drive us and drive AI to make greater things happen.
I'm going to bring up things that I didn't discuss because they're a bit hypothetical, but you already heard my point about this. But I think hibernation and anti-aging are going to be, we're on the verge of, hibernation is a bit far, but there is some progress there. And definitely on the aging research, I think humans are going to be able to stop aging at some point. And I think it's going to be something I was going to bring up in your story. I think this is the element that will change entirely your narrative, because you're assuming that there is a lifespan, but I'm assuming that humans are actually going to live for a very long time very soon.
I'm really excited about the cellular biology. So, for example, stem cells proliferate much more rapidly in space, but then they also kind of like exhaust themselves and burn themselves out. We're learning a lot about, tumor behavior, like cancer cells can become more aggressive. And if you think about the way that we study most cellular biology on Earth, we always put stuff in a dish, right? And the cells stick to plastic. Like, that's not what we're doing with our cells here. And so the fact that you can have them floating, you can do self-organizing structures, you can start to look at really delicate tissue, but you can grow it very big. And it seems like we've talked about a lot of science today, but when you actually add up the number of experiments that we've done, I mean, you can count the tissue chips that we've done on space with my hands and my toes. So compared to the bulk of science that we've done on Earth, the amount of science that we've done in space, it's still possible to read all of those papers, in like a week. You could probably get through a lot of the space literature. And so that just gives you a sense of how much more there is to go. And if we have found this many interesting things with this few number of experiments, I think our hit rate is going to be really high of novel mechanisms, just absolutely undiscovered areas of biology. Because if these were rare things, we wouldn't have found much at all interesting with the small handful of experiments. But the fact that we've just found these wild things that people would not have thought of by sending experiments to microgravity, I think it's going to be huge as we do more and more of these.
And I'm particularly excited about all of these technologies that this incredible team of luminaries is talking about, and the space community and science at large in these avenues, figuring out how to move them through kind of what's called the Gartner hype cycle, right? Where it's novel and it's exciting and it's at its peak and people have this flash of excitement about this piece, and then you could also argue the space industry as a whole for the public has sort of fallen into this trough of disillusionment with it. But out of that comes this slow, steady climb that is ubiquitous adoption. And the dream that I see when I hear these incredible technologies being discussed is, at what point do they become so ubiquitous and adopted that they're just mundane, that we don't see them anymore? And I grapple with the idea that a lot of the general public has no idea that there are people on a space station orbiting around our heads right now. And that both breaks my heart, makes me very happy, because it has been so normalized that you don't think about it. So how do you find that balance between how extraordinary and special these things are, but them being adapted to such a way that we take it as granted as we do with our cell phones?
So I think that's a good transition to our next point. You can start with you and you come back down. So how does all this translate back to space biomedicine? Because we know, for example, from the Apollo era, a lot of things that you use every day, from like your glasses, was the scratch lens on the glasses was actually invented from, the shielding to defend space debris. So you're wearing kind of things to defend space debris. It's kind of like that, right? So what other things that are now currently people don't really take for granted that are being translated to clinical research because of our bridging from Earth to space, right? This is the title of it. And like, what other kind of developments that could help, like which Sylvan, other folks touched on, Kate, Chris, about the aging and other factors, and what are the challenges then in the future to be able to derive that, like what we're doing in space to accommodate what you talked about, Dan, because we come back to space. So sort of you, Sarah.
All right. Well, I will stay in my lane, which is culture, and say that yes, it's so resource strained that in any sort of design thinking, you're designing for the furthest use case. So you're going to have to solve for some very, very difficult problems there first. And then the ability for that to proliferate to the rest of us is so much easier than the relatively comfy scenario that we have in a typical wet lab here at a university like this, I imagine, right? And so figuring out how to leave room for that to kind of trickle down and again become as ubiquitous as we're talking about, that's sort of the magic, and we know we've made it.
So I'm really excited about hardware development. I'm a bench scientist, genomics person, and then I've been at NASA for 16 years, and they totally turned me into an engineer because all I want to do is build stuff now. I think we're talking about all these new commercial space stations. All of them are going to require hardware, and they're there for research, right? They have a few different customer bases. There's going to be commercial spaceflight participants. There's going to be, a lot of new astronauts. There's going to be countries that don't have a space program that want to send their astronauts up there. But underlying all of that is going to be science and discovery, basic research, and then manufacturing and production. And to do any of that, you need equipment. And so we have this amazing International Space Station, but we build our equipment in like dribs and drabs and kind of send a few pieces up, maybe every 6 months or every year. So if we have more than one space station and we're really broadening the scope of who can participate, and now commercial entities are trying to make a product in space or use space to develop their processes, we're going to need a lot of this equipment. And so I think if we are smart with our equipment design, as we're designing things for these space stations, and I talked about this in my talk, I think we can be designing them for Earth as well. Like, we shouldn't design anything to get sent to space without thinking what's the Earth application of this hardware that we're developing as well.
Yeah, so hardware is really, I agree, I think is one of the most applicable thing, and I think self-driving system are going to be really critical, so I brought that up. But I think something else that we didn't discuss that I have high hopes for is more like using space for human on Earth. So for instance, bioprinting in space, I think will have incredible application, and Kate kind of mentioned this. A cell will not fall, it will stay in space. So when you do bioprinting on Earth, you have to fight gravity, right? So you have to put all kinds of like material with your printing to make it stick because it's flat. There's a reason a baby and embryogenesis is happening in a liquid. It's because it allows this three-dimensional structure to be built. And so space provides a unique environment that could really help us, build specific organs for people. Imagine someone who gets their heart printed in space. You could basically create copies of some of our organs in space, something quite unique, could be quite lucrative too. And instead of waiting for a donor, you actually, and you can actually go beyond that. Let's say you have a genetic problem that is linked to a malformation of some kind of organs. You could actually imagine that we first fix with CRISPR engineering. You can change the genetic of that one person, IPA, you create the stem cells, you go into space, and you build now the organ that won't be rejected for that person, because it's still their organ, but improved and built in space, brought back on Earth. So I would say that would be a very futuristic view of the usage of space for some human Earth application.
I always look to access, to do all these things, to take hardware to orbit, you have to have a destination. We have to build that. Right now, the cost to orbit, the time to iterate is far too long. So all of these companies that you've seen and heard mentioned today that are building destinations are betting that you will use it. So that puts you kind of on the hook here, that it's not just use it or lose it, it's a use it or don't have it. So they're going to put it there, so it's going to be expensive early on, but that adoption of use will bring the costs down. So some brave institutions will have to lead the charge to make that happen. And access, iteration have to come down for all these advancements to really start to snowball.
Two things that I'm excited about. One, I think that we're living in a really unique moment from a commercializable standpoint. There's a lot of investment money that's interested in space. We're seeing a lot of interesting verticals coming out of that translational science or space translating into other market verticals, which is very, very, very cool. We have a couple of products that are going to come out of stealth this year that we'll be able to talk about entering the longevity market or the aesthetic medicine market, the surgery market, all derived from with space intentions, which is very cool. That's all money that will feed the continued growth of the space ecosystem, which is all what we want to see in order to make everything happen that my fellow panelists are talking about. But the real reason, the thing that I'm most excited about and the reason why I am connected with all of these amazing people is I see a tremendous amount of lacking in space medicine, in the in the treatment of health and biology of humans in space that I think we, as on Earth, would be shocked about. The fact is, like, the Human Genome Project is 35 years old, the project itself, and Chris talked a lot about how we've used the whole genome sequencing and these short reads to understand human health and biology and disease. Sylvain did a really amazing job talking about precision medicine, and those knowledge graphs are fascinating. But if you look at the way that we talk about medicine today, there was that one image that showed the human and there were like the layers. Medicine takes care of the human at the top of that layer. It's all from disease. It's all from perturbation of health. And in space, it's no different. In space, like, space medicine is built on acute care. It's built on, if there's something wrong with you, then we'll treat it. It's always based on mission dependencies. So if it's affecting the mission, then we need to know about it. If it's not affecting the mission, then it's up to you to tell us. So the people on stage with me, Afshin and Chris, have really led the charge in molecular understanding of human biology in space and moving precision medicine off of planet Earth and into low Earth orbit. And I think that that's what I'm most excited about.
Yeah, definitely excited by everything that was brought up already. And I think also to hark back to some of the medicine questions, most medicines have to be manufactured in space because they, as you probably look when you look in your bathroom mirror, most of the medicines expire within 3, 6 months, maybe 12 months. And if you're starting to look at really long missions, it's an exciting challenge, but also opportunity of having to manufacture what you'll need in long-term missions, either through small biofactories or some small processing that you do chemically. So we'll have to make the things that we're bringing with us or have organisms do it for us, which I think is a fun challenge, a very hard challenge, but it should be interesting. And then also, I think a little bit about point-to-point biology, where we see something weird on Venus or the moon, and we sequence it there, and then send the data back to Earth to do a lot of high-throughput screening to figure out what it is, and then send it back to the planet to do further follow-up work. So I like the idea in the future of a point-to-point biological and biochemical analysis and medicine and experiments.
Yeah, definitely. I think a lot of us are looking forward to using some of that longevity and disease correction here on Earth. So that's great. I love all that. I want to throw something else out there. Perhaps all the space medicine will help us on Earth, but not in space. We might make it to Mars and decide that's the end of it for us as humans. And we may evolve to some kind of post-biological intelligence. How many of you use AI on a daily basis? ChatGPT. I mean, everyone actually uses it, right? Amazon, it's all in the background. It's all happening. And supposedly 10% of all energy used on Earth is used by computers for AI. Ultimately, we'll have to send our computers to space. Not only do they need more energy, like they could orbit the sun and use solar energy, but they have to dissipate heat because all power is eventually lost as heat. So you could see a future, not just thinking of Neuralink, but all of us melding, and that could be a couple hundred years from now. It's something totally different. So I just wanted to put that thought out there for space biology.
That's great. Yeah. So I mean, something related to what you guys are saying, like what are some of the most promising countermeasures that are being developed? 'Cause obviously, sending our dean up to space, is one of the goals we have. And also everyone here, safely, right? But before we do that, you've learned, today, like there's a lot of hurdles we still have to do to make it very safe for all of us to go to space, not just Kate and John, but everyone else. So what are the most promising countermeasures that you think are being, like with
I'll just give a quick one. My countermeasure is to stay right here on Earth.
I mentioned some in the talk, but the Chemferol is one that we're working on, for example, small molecule inhibitors. There's a lot of FDA specific drugs that we've tried on a recent plate where you can put all the various drugs and see what they do in response to spaceflight. So I think epigenetic reprogramming is an option that's down the road, but it certainly in vitro is working. So, but I think we've just really started looking, I feel like, at a lot of the small molecules. So there's probably a lot that would be helpful we haven't found yet.
I'm gonna pass. I don't know. I feel like I don't think we've done enough. To be honest with you, I think that there's so much that we've done to survive in Leo, and I think that we're pretty good at that. I'm not sure, like, for any real consideration of habitation on like the Mars or on the Moon or Mars. I think there's a lot we need to do.
Oh, I fully agree. We haven't done, but we're starting in the right direction. And I think some of the aspects that are in your book, I fully expect that in the future, a bit further out, we'll have Mars humans and we'll have Earth humans. We'll have to, because the environments will be so diverse that as we know ourselves today, and that will come about through epigenetic modifications that are necessary, and maybe they happen prenatal, and people make an election. You want to live on the moon or you want to live on Mars? So I think that's a societal change that will happen, from accepting things as they are in a future reality, which don't exist today. But the technology, I think, is evolving to that.
I think it's going to be hard to have a countermeasure for space, and actually the best countermeasure for me is probably artificial gravity. And there's some hopes there because, and this is where the biology can come and help, is that there's some studies that shows that you don't necessarily need 1G to fight the loss of bone. And it's unfortunate there's not more funding there, but there was something at the Biological and Physical Science Division had pushed a few years ago, Gravity as a Continuum as a program, trying to understand exactly what is the soft spot on gravity where you start losing the benefit of gravity. And there's some recent studies that have shown that actually you don't need 1G, you probably need less. Moon is probably too low. Mars might be actually a soft spot where you can still benefit from the gravity. So once we understand this, you don't have to do a 1G artificial gravity in space, which then simplifies a lot of things. So a lot of research needs to happen there, and this could be a good countermeasure. Radiation, I'm not worried about radiation. We can deal with this, and the antioxidant, and it's great. But microgravity, it's a tough one.
Yeah, I think when we talk about countermeasures, a lot of times, at NASA, people are thinking of like a pill you take or an exercise device for bone. I think we need a countermeasure for space adaptation and how long that takes and how challenging it is. And John knows exactly what I'm talking about. When you get up to space, all of a sudden you turn into a 2-year-old. You don't know how to locomote. You can't even stabilize. There's no such thing as sitting down in space. You're moving all the time. And when you go to a laptop to type your first email home, you type, and then a minute later you end up on the ceiling because you haven't hooked your feet in underneath to stabilize. And so you really learn about every single kind of force that you put into it, and you have to, it's like learning how to play a new sport. You have to learn how to stabilize yourself. You have to learn how to fly. None of us know how to launch ourselves horizontally without putting in some kind of like directional axis. And when you do that, usually you launch yourself up a little bit because you're used to throwing a baseball, and then your forehead hits the hatch. And so I think we need the countermeasure of training and helping people adapt to space, and then when they come back, it's equally as horrible. You're really, there's a lot of neurovestibular dysfunction. I think we can learn a lot about balance disorders and how people sense their body. That's got a lot of, hopefully, potential for geriatrics and aging and all these kinds of neurovestibular disorders. But I think we need to train people more quickly. At least on the coming home point, NASA now has a device that they can put stimulation behind your ears. And they put that on me and dialed it up, and I was like, I feel like I just stepped out of the Soyuz capsule. The lack of coordination, the neurovestibular issues I had, and so they could do that immediately and train me in how to adapt to that. The best thing we have for going up right now is to do parabolas in a zero-G flight, but I think we need to work on how do we get people adapted to space, so when they're going up there for 10 days, they arrive and like they're ready to hit the ground running.
It's a matter of hearts, minds, and wallets all lining up, which is a difficult feat to pull off, and historically takes a war. So barring that, and hopefully not that, making sure that there's a level of investment from public buy-in and commercial buy-in and sovereign state buy-in that gives us the level of funding and support that's necessary to pull this off. Because as much as I want to say and do believe in my heart of hearts that it's the exploration that matters and everything we're going to find up there along the way, if it doesn't get funded and have attributable results, we talk all the time about all of these incredible technologies that we use every day that came from space, but they weren't firmly attributed, at least not from the public's perspective. So whether it's like the Velcro or the GPS or fill in the blank of whichever the tropes you're going to reference, it's ISS is still operating at an annual deficit, and if it were attributed that these technologies that came out of it went back immediately into the coffers, then it wouldn't be. But how do you tether those narratives together and let people understand that the reason why is this?
So that's good. Leads me to, maybe start with Savi, this one, but then we go back around whoever wants to take it, and not everyone has to answer if they don't want. But like, so the collaborative efforts, like John brought up the Axiom Spaces. Axiom's a great company that's really doing great stuff. We're trying to partner with it. And Savi's doing other stuff. Also with the financial aspect, because to do all this stuff and countermeasures, we need money to do it. Of course, as Chris and others pointed out, the NASA budget's might be getting cut a lot this year due to everything that's happening. So like, how would, let's say, filling these gaps in, and how would you go across? I think it's critical for academia to partner with people like Axiom, people with BIOS, or people like with VAS and all these other companies, and same with the government agencies that come along. But how do you guys propose to keep
Well,
these gaps?
I think one of the best ways that academia can publish or can participate and be part of this with companies like BioAstra and with larger commercial entities like Axiom. I had a mentor who always used to say that no one was cured by a PDF. But so much of academia is run off of PDFs. It's run off of really great publications that you guys all shared today. It's run off of the grant cycle. But that's not actually making its way necessarily into people's lives and into the technology. So per Danica's last statement, we have to get faster about that translational cycle and being able to attribute where those discoveries are coming from, how they're being made, so that the general public, the investors, understand the importance of making these investments and making them early.
Anyone else want to chime in?
Yeah, I think we need to do a lot in terms of efficiency. It's very expensive to get to space. It's very expensive to do your experiment in space, and there's the reason for that. It's not a good reason. It's just that historically, NASA flew shuttle flights, and to fly in a shuttle flight, you have to build a shuttle mid-deck locker, which is kind of a shoebox-sized thing. They didn't have bench tops on the shuttle. They didn't have lab equipment that was standard, and we carried that with us through to the International Space Station. So when I started in 2009, all that lab equipment that I showed you, we've brought up there since then. And so getting them to understand that it's not just the structure on the outside of the space station and providing the power, but the lab equipment. So right now, if you're a researcher and you want to send your experiment to space, NASA goes, great, build the centrifuge, build the cell culture vessel, build the machine that's going to adjust your temperature and CO2. And so you have to spend 5 years building your hardware to get your experiment done. This is crazy. Like, it's just nuts that we're doing this way. And so I think we've got a chance with the commercial providers to have them build the station, and either they will or another kind of a third layer will come in between the commercial stations and the scientists to build that equipment. And so that we can have standardized equipment that there's going to be this sequencer up there's going to be this PCR machine, this centrifuge, and you don't have to go build the centrifuge before you can do your experiment. And so that's going to bring the cost of your experiment way down. There's going to have to be some fundamental investment, whether that comes from the companies, the governments. That's a slice off the top of everybody who's going to these stations and paying for it. So there's going to have to be overhead, but we need to build that equipment, and then it's going to be so much cheaper because instead of flying, everything is costed in terms of kilos to orbit, dollars per kilos to orbit. So you don't have to fly all this heavy equipment. All you need to do is fly your tube of cells or fly your particular drug that you're adding. And that will bring the cost down into something that like every researcher here could completely reasonably afford. And that time scale has now been shrunk to, a few months to do the safety review of your particular substance. All the hardware has already been approved.
So I think,
I'd like to see universities, especially the STEM portions, do more design and build classes. This takes an incredible amount of time and effort from the professor. Even though you have fewer lectures, you have more hands-on. It helps train our workforce, and we can get sponsors from industry. And then these undergrads, they're primed to help with research in the summer. So admittedly, that's not going to replace your army of grad students, but the undergrads can do a lot. And I've personally taught a class like this at MIT. I've got a son who graduated from Worcester Polytech, a school that's designed entirely around hands-on projects, and I think that's what we need to do going forward.
that's great. So in our last minute, I want each of you guys to talk about in one sentence what keeps you up at night for space science and what excites you. So it's hard, one sentence. So keep it short.
The possibility of life beyond Earth, even really close on a neighboring planet.
The crackling, crunching noise of the Earth being enveloped by the sun.
wondering what Chris Mason will say on a panel. And funding. Funding is what keeps me up at night.
The fact that I may not be able to go to space again.
We'll get you up there.
Yeah, we'll get you.
What keeps me up at night? I don't know, not much. I guess I'm very focused on longevity and kind of the future of mankind. That's what keeps me up at night.
I think about all of the discoveries that are yet to come and making sure that we enable the architecture and the infrastructure to do that.
The inevitable extinction of our species and delaying that as much as possible.
So thanks. Let's thank the panelists and our team. Close us out.
Oh, you can stay right there. We'll just, so thank you again. This has been an amazing morning. I think I've learned more in this 4 hours or 5 hours than I have in years of reading science fiction and years of reading books about space. So thank you, and this was very inspiring as well for all of our researchers, and hopefully for all the great things we'll all do together. One quick question for all of you, or a suggestion, I should say. One of the things that we should think about as many groups are doing various space exploration and space space stations, we should build a public-private laboratory there. And some key university partners and some key commercial NASA as a consortium, we should have a zero gravity research institute that should actually be doing a lot of these cool things in a collaborative way. And then it won't be anyone's particular cost or domain. So you're the people, are all the right people to make that happen. So let's all work on that together. But thank you again. What a wonderful day. Thanks for every one of you coming here to Pittsburgh.